Acheter une truc ici. Après ça, j'ai gardé le perçoit, j'ai déjà même
c'est pas
tu ne verras pas les ça c'est mieux par rapport à tout ta diapositive. Donc maintenant, ce que tu fais, moi, pendant mes questions, c'est encore tout le truc.
Mais on est mis
alors, quand ça se finit, en fait, et toi, tu auras fini tes questions, je suis en train de faire un rotage, je vais revenir sur la formation. Dans ce cas, tu viens et tu là, tu es sur ta diapo, tu fais Alt Tab et tu fermes.
ouais, quand c'est fini, là.
On est bon? Moi, je vérifie.
Il est là, regarde. Il est là. Ouais. Il va falloir que je vienne un petit peu regarder. Ah, je vais prendre la version.
Merci tout le monde, c'est super qu'on a une salle bien pleine. Est-ce qu'on a le son sur la visio, tu sais?
Je crois que oui.
Ok.
Oui, parce que quand je parle, il y a la visio.
D'accord. Donc aujourd'hui, je vais présider le jury de HDR de Monsieur Frago, Henrik Frago. Et donc on a 3 rapporteurs et 3 examinateurs et examinatrices. Juste après moi, je vais laisser que chacun d'entre vous se lève et se présente brièvement. Frago va faire sa présentation. On va laisser une petite pause pour que ceux qui veulent partir avant les questions puissent partir. Et ensuite, il y aura à peu près 15 minutes de questions par membre du jury. Quand on arrive à 15 minutes, je vais vous signaler pour une dernière question. D'accord? Donc, si vous voulez... Ah, je me présente. Donc, Christine Ménard, directrice de recherche INRAE au CBGB.
Bonjour, toutes et à tous, Paul Delta, je suis chercheur au CNRS, je travaille donc à Lyon au laboratoire biométrie, biologie et véhicules, et je m'intéresse pour le temps aux interactions entre arthropodes et bactéries symbiotiques.
Bonjour, moi je suis Elodie Bertrand, je suis à l'INRAE de Sophia Antipolis et je travaille sur la dynamique des populations d'insectes et des
Jean Bourgard, je travaille à l'ERLAC, c'est le Swiss Federal Institute of Aquatic Science, et j'enseigne à l'ETH Zurich dans les domaines de l'écologie et de la biologie évolutive.
Bonjour, Nathalie, je suis chercheuse à Paris, basée à la Sorbonne Université à Jussieu. Et je travaille principalement sur les réseaux d'interaction entre espèces avec pas mal d'approches de modélisation.
Bonjour, je suis Alison Duncan, je suis ISEM et je travaille sur les interactions entre parasites, l'inclusion de la virulence, et je travaille avec les virus de plantes et des acariens.
Bon, c'est à moi. Bon, merci beaucoup à toutes les personnes pour être venues aujourd'hui. Merci aussi à les personnes que vous êtes en ligne à cette HDR qui s'intitule A Multitrophic Approach to the Dynamics of Insect Herbivores, Experience with Plant Predators and Insect Symbionts. Moi, je m'appelle Frago, je suis chercheur au CIRAD et je travaille ici au CVGP depuis 5 ans. Tout d'abord, un grand merci à les membres du jury d'être venus en présentiel. Ça, c'est plutôt exceptionnel aujourd'hui. Merci Elisa, Elodie, Fabrice, Alison, Christophe et Christine. Et je vais commencer pour les remerciements. Un nombre plutôt long de personnes qui ont fait possible ce jour aujourd'hui. J'ai marqué en vert les étudiants en master que j'ai encadrés, en bleu les étudiants en thèse que j'ai interagi, et en rouge les étudiants en thèse que j'ai co-encadrés, au passé, maintenant avec Francisco. Pour commencer avec les détails des remerciements, un grand merci à beaucoup de personnes dans ma vie, les amis, la famille, mon fils, ma compagne Clara, des collaborateurs comme Dirk Sanders, que ça fait depuis la fin de ma thèse avec qui je travaille de manière très intense. Un grand merci aussi à toutes les personnes que j'ai trouvées pendant mes 2 postdocs aux Pays-Bas et en Angleterre, où j'ai commencé à encadrer un nombre important d'étudiants en master, et ma première thésarde de l'Aia Fontana en collaboration avec mon directeur de thèse, Jesus Selfa. Après ces 2 postdocs, j'étais embauché pour aller à l'île de la Réunion comme chercheur CIRAD, où j'ai rencontré beaucoup de personnes que je vais remercier aujourd'hui aussi. Là, j'ai co-encadré 2 thèses en collaboration avec Samuel Nibouche à la Réunion, Niridi Anzinga et Karim Kriwart. Et finalement, je suis venu ici au CVGP. Grand merci pour l'ambiance humaine et de travail, que c'est vraiment génial. Pas mal d'étudiants en master, et Francisco qui a démarré la thèse, il fait à peu près quelques semaines. Bon, je vais commencer un peu avec le contexte général. J'ai pris le titre de ma présentation et j'ai souligné quelques mots que je vais parler un peu. Pas besoin de définir les insectes herbivores, mais c'est pour vous dire que moi j'ai fait toute ma carrière sur les écosystèmes terrestres et sur ces groupes d'insectes qui jouent un rôle très important dans les écosystèmes terrestres. Il peut moduler la production primaire, il peut avoir un effet sur la diversité des plantes et même sur la minéralisation des sols. J'ai marqué aussi le mot Dynamics and Experiments parce que la plupart de mon programme de recherche, c'est un programme de recherche d'écologie expérimentale des communautés qui s'oppose un peu à l'écologie observationnelle où on regarde la diversité qu'on a dans les écosystèmes pour comprendre cette diversité. Dans l'approche expérimentale, on prend cette diversité, on la met au laboratoire, on la manipule, et comme ça on arrive à avoir une approche un peu plus mécanistique, comprendre comment ces espèces ils interagissent. On peut par exemple suivre la dynamique de ces communautés pendant plusieurs générations. C'est ça qu'on appelle les long-term multigenerational dynamics. Et ça peut nous donner des éléments pour comprendre la diversité ou même les extinctions. Pourquoi une espèce n'arrive pas à rester dans un système parce qu'il est par exemple disparu par la compétition par exemple. Multitrophique, en définition très simple, c'est une interaction qui implique plus de 2 niveaux trophiques. Je vais parler un peu plus de ça avec un exemple d'un réseau trophique. Les plantes qui sont nourries pour les herbivores et attaquées pour les ennemis naturels. Pour le reste de ma présentation, quand je parle d'ennemis naturels, je vais parler des prédateurs et parasitoïdes. Et quand je parle de biocontrôle, même si le concept il est un peu plus général, je vais parler de la suppression des ennemis naturels sur les herbivores. Je vous montre ici un guêpe parasite, que c'est un groupe que je vais parler, un parasitoïde. L'adulte il va déposer un œuf à l'intérieur ou à la surface de cet hôte qui va se développer, il va tuer l'hôte, il va devenir un autre adulte. Dans ces réseaux, on voit pas mal de complexité déjà, on voit les liens d'interaction, mais moi je vais vous parler d'interactions un peu plus complexes que ça même, parce que des fois, il y a des interactions qu'on ne voit pas dans les réseaux. Et c'est par exemple le cas de la prédation intra-guilde, que je vais parler beaucoup aujourd'hui, que ça arrive par exemple quand 2 ennemis naturels ils partagent un même proie, mais ils s'attaquent entre eux en même temps. Ces guêpes parasites, ils peuvent déposer un œuf à l'intérieur de ces pucerons, le puceron est consommé par la coccinelle va se nourrir de manière indirecte de ces guêpes parasites. Alors on voit que les niveaux trophiques ne sont pas vraiment fixes. On voit ces guêpes qui arrivent à surmonter un peu, ils montent un peu en haut sur ces chaînes alimentaires, ils deviennent un prédateur des prédateurs. Et j'ai publié un papier il fait quelques années qui a déclenché un peu ces lignes de recherche sur la prédation intraguilde. Si tout ça c'est déjà compliqué, si on rajoute les microbes, ça devient encore plus complexe. J'ai travaillé beaucoup sur la symbiose chez les herbivores. On a publié un review qu'on parle de les différents services que ces microbes associés à les insectes herbivores ils peuvent donner à ces herbivores. On a pas mal de symbiotes qui vont donner un service nutritionnel, même un effet sur le comportement. Il y a même des symbiotes défensifs que j'ai travaillé un peu, qui vont protéger ces insectes contre les ennemis naturels. Je souligne ici un groupe que c'est les tisanoptères, les thrips. Ce n'est pas un groupe très très connu, mais que je vais parler beaucoup. C'est un groupe c'est un ordre d'insectes qui est au même niveau que les coléoptères, les pédoptères, les papillons et les escarabées par exemple. Mais c'est beaucoup moins divers et c'est très petit et on ne le connaît pas toujours très bien. Pour vous mettre un peu de jolies photos, hein, ça c'est une photo de Bruno Michel. Ça c'est un insecte qui fait 1 mm, c'est tout petit. Des fois, je ne sais pas si je travaille avec les insectes ou la microbiologie. Ben, ça c'est un thrips, ça c'est un acarium herbivore, un tétranique, et c'est aussi très petit. Il y a même des thrips prédateurs. Il y a des prédateurs de thrips spécialisés, comme cet acarium prédateur aussi. Les thrips sont importants, ils peuvent donner lieu à des gros dégâts dans l'agriculture. Ils peuvent même transmettre des virus. Vous les voyez, ces petits points marrons, c'est les thrips. Ils peuvent passer des virus des plantes qui vont causer des dégâts sur les cultures. Bon, après cette introduction, je vais parler un peu des axes de ma recherche. Le dossier écrit qui est là, il a 3 chapitres. Un chapitre des interactions avec les plantes, les interactions avec les ennemis naturels, toujours avec un focus sur les herbivores, et les symbiotes. Merci à Martha, à ma cousine, qui a fait ces aquarelles exprès pour l'HDR. Mais aujourd'hui, je vais vous parler que de ces 2 axes. Et je vais me concentrer sur les projets vraiment récents, des projets qui ne sont pas encore publiés et dans lesquels j'ai commencé mon encadrement à superviser des étudiants en thèse et en master. Et on le voit ici, je vais entrer dans les détails après. Pour commencer, pour le premier axe, c'est la question des interactions complexes entre les ennemis naturels. J'avais commencé à s'évoluer un peu quand j'étais recruté à la Réunion. Il y avait une forte demande sociétale pour la gestion de ces insectes, les thrips et les acariennes, chez la production de poivrons. Donc, vous voyez ici un serre expérimental de poivrons où on mettait des petites microcobes, qui mettaient des combinaisons d'ennemis naturels à l'intérieur. Ça, c'était un volet que j'ai fait en collaboration avec Samuel Nibouge et avec qui on a encadré 2 thèses. Ça, c'est la petite équipe. On voit ici Niri. On a même fait une mission à Madagascar pour récupérer des thrips avec cette micro-caméléon. On a fait beaucoup de manipes avec Karim, même dans les serres de rosier expérimentales. Et Laure qui a fait le master aussi avec nous. Bon, l'idée principale de ces volets de recherche, c'est plutôt de comprendre la relation entre la biodiversité et le biocontrôle. On sait par pas mal d'observations que quand on est dans un écosystème où il y a beaucoup de diversité d'ennemis naturels, la suppression des herbivores, le biocontrôle, il va être supérieur. Ça, c'était très clairement démontré avec ces méta-analyses, où les auteurs ont pris des données de toute la planète, ils ont trouvé cette vraiment jolie corrélation positive entre ces 2 variables. J'aimerais avoir des corrélations comme ça des fois, parce que ce n'est jamais le cas. Mais bon, il y a beaucoup de données. Et cette corrélation, à niveau mécanistique, on parle ça émerge à partir de la complémentarité des niches de la facilitation. Ça implique qu'on a 2 ennemis naturels, même si on a une cible d'un herbivore, ces 2 ennemis naturels vont se complémenter, ils vont attaquer chacun un stade différent de cet insecte. Et c'est là où on va éviter de compétir fortement, ils vont partager leur niche, et ça va être positif pour le biocontrôle, parce que chacun il va attaquer un stade de cet herbivore. Mais pour moi, cette corrélation, il implique un petit paradoxe, que c'est que dans les écosystèmes très divers, comme les écosystèmes tropicaux, il va y avoir beaucoup d'ennemis naturels qui vont s'engager à des interactions antagonistes, comme c'est lézard qui va être un prédateur spécifique des autres ennemis naturels ou ces guêpes qui va attaquer un autre guêpe. Et c'est là où on a exploré un peu cette dualité, surtout avec notre travail, avec la prédation intraguilde que j'ai déjà parlé avant. C'est pour ça que moi j'aime bien de représenter le biocontrôle comme ça, avec ces deux axes, l'axe des interactions positives entre ennemis naturels négatifs, comme la complémentarité et le biocontrôle. S'il n'y a pas de prédation intraguilde et beaucoup de complémentarité, on espère qu'il va y avoir un bon biocontrôle, et l'inverse, quand les conditions sont à l'autre côté. La première manip qu'on a mis en place, c'était pendant la thèse de Niri. On a mis en place ces manips avec ces 4 espèces d'arthropodes, 2 espèces de thrips, Franklin occidentalis et Thrips parvispinus, et 2 acariens prédateurs. Les noms sont plutôt compliqués, alors je vais représenter pour le reste de ma présentation comme ça. Le thrips jaune, l'acarien jaune, le thrips rouge, l'acarien rouge. Désolé pour les taxonomistes, je sais que vous n'aimez pas ça, mais c'est pour vulgariser un peu. Ce système il était intéressant parce qu'on le trouvait que ces 4 espèces ils coexistaient dans les serres des productions agricoles de poivrons. Et c'est un système intéressant parce que les acariens s'attaquent entre eux, c'est un prédation intraguilde réciproque, ils peuvent s'attaquer un à l'autre, et les acariens peuvent attaquer les deux thrips. En plus, on a commencé à collaborer avec un biofabrique qui produisait ces ennemis naturels, qui avait mis ces 2 espèces en élevage expérimental pour les produire en masse et les lâcher dans les serres pour le biocontrôle. C'est là où on a commencé à interagir pour optimiser cette moyenne de lutte. Bon, les hypothèses, on a émis des hypothèses, d'accord avec la théorie, quand on a les 2 ennemis naturels, on atteint un peu plus de suppression chez les herbivores, mais on voulait mettre aussi une hypothèse par rapport à la coexistence, et on va dire que pour coexister en bonne manière, ça serait que chaque acarien il se spécialise sur un de ces thrips. Regardez les flèches maintenant. Là, c'est notre hypothèse, qu'ils vont se spécialiser, il va y avoir une réduction de compétition, même si entre eux, ils vont s'attaquer. D'accord? Bon, pour mettre en place cette manip, c'est une approche classique qu'on fait encore aujourd'hui ici au CBGP. On a ces cages de 40 cm, un volume comme ça. On met les plantes à l'intérieur, on installe les ennemis naturels et les herbivores, et on fait un suivi temporaire. Et la seule chose qu'on fait chaque semaine, c'est compter. Bon, la seule chose, ça c'est compter, c'est la partie plus complexe. Hein. Il faut compter, et on rajoute des plantes. Mais on ne manipule pas une fois on a lancé la manip. Pour ces manips en particulier, on a mis toujours les 2 thrips dans tous les cages, et des cages témoins sans ennemi naturel avec un ennemi, l'autre, ou les 2 ensemble. Et on a suivi la dynamique pendant presque un peu plus de 3 mois. Ça veut dire que si ces arthropodes ils ont un cycle de développement d'entre 5 et 7 jours, on a suivi au moins 10 générations de ces espèces. Et là, on peut parler de long-term dynamics, extension, coexistence, etc. Bon, ici, les résultats. Je représente ici les résultats pour un thrips et l'autre. En vertical, vous voyez la moyenne d'individus par centimètre carré de feuilles. Parce qu'on avait fait à mesure qu'on sortait les feuilles pour faire le comptage, on mesurait un peu la feuille et on comptait. Et ça, c'est compacté en tous les 13 semaines, mais les analyses sont pas faites avec un simple ANOVA, c'est des modèles qui considèrent la corrélation temporelle, par exemple. Et vous voyez 4 barres, un, le témoin, avec un acarien, avec l'autre, ou les 2 ensemble. On voit que pour le thrips jaune, et peut-être un peu d'accord avec notre hypothèse de la spécialisation, on voit que l'acarien jaune, il n'arrive pas à faire une décroissance significative de la densité de cette espèce. Mais par contre, l'acarien rouge, Par contre, quand il y a l'acarien jaune, seul ou en combinaison avec l'autre, on voit une décroissance. On peut dire qu'il y a cette hypothèse de la spécialisation, mais si on voit l'autre espèce, on voit que ce n'est pas le cas, parce qu'on voit à nouveau, au témoin, la densité c'est supérieur, il y a plus d'individus par feuille, par centimètre carré de feuille. Les autres acariens seuls, ils décroissent la densité, mais pas significativement. Et quand on a les 2, là, il y a une suppression significative. Là, on pourrait parler que peut-être ces 2 acariens, ils se complémentent pour arriver à réduire les densités de cette espèce. Un donné que je trouve très intéressante, c'est de regarder la densité de ces acariens prédateurs. Ça, c'est pour l'acarien jaune, et ici, l'acarien rouge. Et on voit le même, hein, la densité de ces espèces par centimètre carré de feuilles, quand l'acarien est seul ou en situation de prédation intraguilde. On voit que l'acarien jaune, il va toujours très bien, il n'y a pas d'effet de la présence de l'autre acarien, de compétiteur, Mais l'acarien rouge, il souffre énormément de la présence de son compétiteur. On voit une décroissance vraiment très importante de ses densités. En fait, dans ces manips qui a duré 13 semaines, à partir de la semaine 7, quand cet acarien il est en combinaison avec l'autre, il a disparu. On a enregistré des extensions dans notre cage. C'est là où on peut dire que cet acarien, c'est un compétiteur supérieur par rapport à l'autre. Il n'y a pas de coexistence. Dans notre cage, parce que là, c'est des cages où l'espace est très limité, il n'y a pas la possibilité de s'échapper, de trouver des refuges. Peut-être dans les serres où il y a plus de complexité spatiale, c'est différent. Mais dans notre cage, il n'y a pas de coexistence. On voulait comprendre un peu pourquoi cet effet, et c'est là où Laure Madeleine elle a fait le master pour explorer ces questions au niveau comportemental. Laure, elle a fait des manips en disque foliaire, que vous voyez ici. C'est des disques de 3 cm de diamètre, comme ça, qu'on connecte avec un petit bâton, et on met l'acarienne au départ d'un disque foliaire, et on compte le temps qu'il va prendre pour aller à l'autre côté. Et dans le disque foliaire d'origine, on fait une série de traitements chimiques pour explorer si ces traitements chimiques ils vont avoir un effet sur la dispersion. Ces traitements, c'était 3 traitements différents. On avait un témoin où on ne faisait rien, un témoin avec l'autre ennemi naturel, le compétiteur, où on laissait l'ennemi naturel se promener dans ses feuilles pendant 24 heures, et après on mettait l'autre, et on mettait le petit bâton, et on comptait le temps pour croiser l'autre côté. Vous voyez les résultats ici. Ici en bas, vous voyez le temps, c'est les 6 minutes de la manip, que le max qu'il donnait à l'acarien pour se disperser. Et en vertical, vous voyez la proportion d'individus qui vont se déplacer vers l'autre côté. Au début, évidemment, ils sont tous là, et à fur et à mesure que le temps il passe, ils vont se déplacer. Et on voit que par exemple, dans ces traitements A, à partir de 250 secondes, la moitié des acariens s'est déplacée vers l'autre côté. D'accord ? Et on voit ici que l'acarien jaune, il s'échappe plus rapidement en présence de l'autre compétiteur. Il évite la compétition. Par contre, l'acarien rouge, le mauvais compétiteur, on va dire, il n'a aucun effet, il se déplace, mais sans sentir l'odeur de son compétiteur. Et en plus, Laure, elle a démontré que cet acarien, il a la capacité de distinguer entre les œufs de lui-même et de son compétiteur. Il va prédater en préférence les œufs de son compétiteur. Alors, et lui, il ne peut pas faire ça. Alors lui, il évite les traces de lui, mais quand il le trouve, il le mange. Clairement, c'est là où l'écologie expérimentale peut nous donner des éléments de pourquoi les espèces ils arrivent à coexister. Ce travail, je le trouvais intéressant parce qu'il y a eu un bon transfert vers l'appliqué. On avait travaillé, comme j'ai dit, avec la biofabrique, la coccinelle. Si on regarde aujourd'hui le site web de la coccinelle, on voit qu'il vend aujourd'hui pour les cerfs toute cette espèce d'ennemi naturel. Il vend encore l'acarien jaune, la Matryoshka, qui c'était le bon compétiteur, mais l'acarien rouge, il a disparu du menu de la coccinelle. Je pense que notre travail, ça a aidé à optimiser ces élevages, à prendre cette décision, et c'est là où l'écologie expérimentale il peut apporter beaucoup à l'écologie, à la lutte biologique appliquée. On a fait un autre manip lié à cette thématique pendant la thèse de Karim Tichiwart, qui était aussi co-encadrée par Samuel Nibouche. Là, on a augmenté en diversité. On a créé un système avec 2 espèces de trips, un tétranique et un puceron. Je pense que si on rajoute ici les alérodes, on a tous les gros groupes de ravagères dans les serres de production de horticoles. Et là, à nouveau, on a collaboré avec la coccinelle, on a travaillé avec un acarien qui attaque ces 2 trips. Ça, c'est le bon acarien de la manip précédente. Pourquoi mettre à l'autre s'il n'est pas très dué? Après, on a travaillé avec 2 ennemis naturels de puceron, la guêpe qui attaque le puceron et la coccinelle qui attaque le puceron, mais qui peut se nourrir de la guêpe comme prédateur intraguilde. Et un prédateur généraliste qui attaque tout le monde et qui peut même se nourrir de 2 de ces ennemis naturels comme prédateur intraguilde. Si on compte les lignes ici, vous voyez qu'il y a 11 flèches. Ça, c'est un de nos variables principales. C'est le flèche, c'est le lien de prédation. Et de ces flèches, il y a 3 qui sont rouges. C'est le lien de prédation intraguilde. Et c'est avec cette communauté qu'on a manipulé, qu'on a joué avec ces 2 variables pour créer un gradient des communautés avec un nombre variable de liens de prédation et liens de prédation intraguilde pour tester notre hypothèse, que c'est la suivante. Et d'accord avec la théorie, on attend que quand il y a plus de liens de prédation, la sous-pression sur les herbivores ça va être plus forte. Et quand il y a moins d'interaction entre guildes, il va y avoir aussi une sous-pression qui va être mieux, parce que l'intra-guilde, on dit toujours qu'il peut y avoir un effet négatif. Si les ennemis s'attaquent entre eux, ça va avoir un effet négatif sur la sous-pression des herbivores. Bon, 2 exemples de comment on a créé ces 8 communautés. Hein. Si on élimine la coccinelle, on finit avec une communauté avec 9 liens de prédation et 2 liens de prédation intra-guildes. Si on élimine le prédateur généraliste qui s'engage avec beaucoup d'interaction, on finit avec une communauté de 5 et 1. On a mis en place ces 8 communautés répliquées 10 fois, 80 unités expérimentales. Ça, c'est la manip la plus grande que j'ai jamais été impliquée. J'ai compté cette semaine, et Karim, il avait compté plus d'un demi-million d'insectes. On les compte tous un par un. Hein. On fait des groupes de 100, 100, 100 et on compte très rapidement. Mais quand même, le comptage, c'est un peu... mais les données qu'on obtient sont aussi très puissantes. Ça, c'est le message, c'est pas que compter, c'est aussi avoir un effet après. Hein. Et on a suivi la dynamique pendant 20 semaines avec des comptages sémanaux. Et si je vous montre un peu toutes les données, il ne faut pas essayer de comprendre ça au détail, mais en général, chaque colonne, c'est une communauté de les 8 qu'on a créées. Chaque ligne, c'est une des espèces d'insectes. Je vais me centrer sur ces 3 herbivores. Vous voyez, chaque graphe, c'est en vertical, c'est la semaine, et vertical, c'est le nombre d'individus qu'on a compté. En horizontal, c'est le temps. Et chaque ligne de couleur, c'est un des réplicants. C'est la dynamique dans une petite cage. Et on voit que des fois, les comptages, ça va à 1500 par jour. Un jour que tu fais un comptage de 1500 puissants. Et là, avec tout ça, on a fait des stades que je vous montre de manière très simplifiée ici, hein, pour tester nos hypothèses. Ici, c'est le nombre, chaque point dans ces graphes-là, c'est une cage, et c'est le nombre cumulatif d'individus qu'on a compté dans ces cages-là. C'est pour ça que les valeurs, il faut monter jusqu'à 12 000. Et en bas, on voit le nombre de liens de prédation, d'accord, pour les 3 espèces. Le premier message, c'est que les liens de prédation intra-guildes, ils n'avaient pas un effet énorme sur la dynamique de cette communauté, à nouveau. Mais par contre, les liens de prédation intra-guildes, les liens de prédation, ils avaient un effet, d'accord avec notre hypothèse, ils avaient un effet positif sur le biocontrôle, ça veut dire négatif sur la quantité d'insectes. Plus de liens de prédation, moins de pucerons, moins de trips, un biocontrôle qui est mieux. Mais on a trouvé l'inverse par rapport à le tétranique. On a observé que quand il y a beaucoup de liens de prédation, ce tétranique, il explosait. Pourquoi? Et ça, à nouveau, l'écologie expérimentale, il nous aide à comprendre ça. Si on voit cette communauté ici, on voit que le tétranique, il est attaqué que pour la punaise prédatrice généraliste. Quand on rajoute beaucoup d'ennemis, là, chez 2, il disparaît. Et c'est ça qui déclenche la densité, l'explosion de cette acarienne. D'accord? Et en plus, on a fait des manips, des comportements qu'on voit que quand on lui offre à ses prédateurs tous ses ennemis, il préfère se nourrir de ces 2. C'est pour ça que là, c'est un fait qu'il y a beaucoup d'ennemis naturels, l'exploitation de la plante, il est moins important, et lui, il arrive à exploser. Un critique important que vous pourrez avoir à ces 2 manips que je viens de présenter, c'est qu'on prend toujours une communauté, qu'on la manipule, mais toutes les conclusions qu'on arrive à sortir sont limitées à ces communautés. On ne peut pas vraiment généraliser. C'est là où je dis qu'on manipule une communauté, mais on n'a pas vraiment des réplicats à niveau de communauté. C'est pour ça qu'on a déposé un projet à l'ANR qui était retenu, qui a commencé au mois de janvier, qui s'appelle Enemy Cocktail, Designing Natural Enemy Cocktails for a Better Biocontrol, où on va essayer de surmonter ces limitations. C'est un travail qui implique beaucoup de personnes au CVGP, surtout à la plateforme CEPA, la plateforme Serre Élevage Finataire d'Arthropodes. Il y a des collaborateurs à l'ISEM, il y a un autre biofabrique impliqué, c'est BioBest, et Dirk Sanders à l'université d'Exeter. La plateforme CEPA, il est en train de chercher un logo. Je profite pour faire un peu de lobbying sur le logo que j'ai proposé. Je n'ai jamais eu une très jolie réponse, mais je laisse là. Et ce projet, il va avoir un parti expérimental important qu'on a déjà commencé avec Molly, qui était ici pendant 3 mois, Amandine, qui est en CDD, Francisco, qui a commencé sa thèse. Je profite pour remercier Anne et Nico, avec qui je travaille beaucoup sur la plateforme. Les manips qu'on va mettre en place, là, on va utiliser le gros menu de la biofabrique BioBest, où on peut sélectionner et faire des communautés répétées à droite, à gauche et vraiment faire un vrai dessin répété à niveau communauté pour tester cet effet de la complémentarité prédation intragile sur la dynamique des communautés. Il y a un côté de modélisation avec ces données qu'on va obtenir. Pour le modéliser, c'est de comprendre à niveau mécanistique encore mieux ces interactions. Et ça va nous permettre de sélectionner les meilleurs cocktails, les meilleures combinaisons et les tester en direct dans les serres d'exploitation agricole. Là, on est obligé de passer en collaboration avec le privé qui va produire en masse ces ennemis naturels. On va chercher des serres répliquées. On va faire ce qu'on appelle engineering the trophic web, où on a des réseaux trophiques déjà dans les serres. On va lâcher ces ennemis naturels avec un, l'autre ou deux et voir comment on arrive à réduire l'impact des ravageurs ou même voir s'il y a une réponse des émergences des ennemis, des ravageurs secondaires par exemple. Pour finir ces parties-là, avec ces deux manips qu'on a fait, on voit que la pression intragile, même si on parle toujours que la pression intragile c'est vraiment le diable de le biocontrôle, on voit avec ces manips que ce n'est pas le cas. Et on a même des manips que je n'ai pas montré, qu'on a même trouvé que la pression intragile, il peut même stabiliser la communauté, il peut nous aider à avoir un biocontrôle durable, que les espèces arrivent à se maintenir dans les écosystèmes. Ce n'est pas toujours nécessaire d'avoir plusieurs ennemis naturels, je ne devrais pas le dire parce que je suis en train de démarrer un projet des ennemis cocktails, mais ce n'est pas toujours nécessaire, des fois il y a un seul ennemi qui fait tout le boulot, on est bien. Et évidemment, les ennemis généralistes, c'est très compliqué de les mettre en place, il faut bien le comprendre parce que cet ennemi naturel, on a vu qu'il avait une préférence pour ces puissements, parce que les tétraniques ils font des toiles et manger sur ça c'est difficile, il faut utiliser des spécialistes. Les généralistes ils peuvent même déclencher les explosions d'ennemis naturels, des ravageurs secondaires. Et l'objectif final du projet Ennemi Cocktail, c'est de définir un roadmap pour la sélection des combinaisons d'ennemis naturels, pour le contrôle biologique, avec des ennemis naturels dans les serres d'exploitation. C'est toujours limité à les serres, à la production sous-aride. Très bien, on va bien au niveau timing, alors je peux me relaxer. Je vais parler de la deuxième partie qui est beaucoup plus courte. C'est sur la symbiose chez les herbivores. J'ai travaillé beaucoup pendant mes périodes de postdoc sur comment ces symbiotes chez les herbivores peuvent avoir un effet sur la plante et sur les ennemis naturels. Mais aujourd'hui, je ne vais pas parler de ça. Aujourd'hui, je vais parler de symbiotes chez les trips. Et comment ces réseaux entre plantes, trips et symbiotes, il peut avoir un changement de communauté à niveau de gradient d'altitude. Je vous présente ici le gradient d'altitude avec ces montagnes désignées pour Montpetit. Et pourquoi on s'intéresse à les gradients d'altitude? Bon, c'est une bonne manière pour prendre, dans une région géographique limitée, on a un certain pool d'espèces. Et on va exposer ces poules d'espèces à un gradient de température, par exemple, sans avoir un changement de communauté. C'est la même communauté exposée à des changements abiotiques. Et ça, évidemment, ça peut nous aider à comprendre qu'est-ce qu'on va avoir quand le planète il va chauffer 5 degrés. D'accord? Alors, on sait bien qu'à haute altitude, les températures sont inférieures. Il y a moins de ressources. Par exemple, les plantes, ils ont moins de temps pour se développer. Il y a moins de ressources. L'unité temporelle c'est plus petit, les conditions de vie sont plutôt difficiles et ça implique qu'à notre altitude il y a moins de diversité. Jusque-là, je pense qu'on est presque tous d'accord, mais qu'est-ce que ça donne au niveau des réseaux d'interaction? Et c'est là où il y a un peu on n'est pas tous d'accord au niveau des hypothèses qui peuvent s'émettre. Vous voyez que quand il n'y a pas beaucoup de diversité, une hypothèse c'est que quand il n'y a pas beaucoup de diversité, les peu d'espèces qui arrivent à se développer dans ces conditions très dures, ils vont devenir très spécialisés parce que les spécialistes, par rapport à les généralistes, normalement sont plus efficaces à exploiter la ressource. C'est là où on voit ces réseaux, ça c'est les plantes en bas, les herbivores en haut, et on voit qu'il n'y a pas beaucoup de connexions. Chaque herbivore il va se nourrir de 1 ou 2 ressources. Ça c'est une des hypothèses. L'autre hypothèse, qui c'est peut-être un peu plus acceptée et que c'était l'hypothèse de départ de notre manip, là, l'hypothèse de départ, ce n'est pas pour le fait qu'on a trouvé ce résultat qu'on a mis cette hypothèse, c'était une hypothèse a priori. D'accord? Et l'autre hypothèse, c'est qu'en haute altitude, les espèces deviennent plus généralistes. Parce que dépendre d'un sol ressource dans une condition difficile, c'est très risqué. Si cette flore que tu vas te nourrir, cette année, il n'a pas fleuri, par exemple, c'est fini. Par contre, si tu peux bouger entre plusieurs ressources, ça, c'est une bonne stratégie en des conditions difficiles. La question des gradients d'altitude, on l'avait démarré pendant la thèse de l'IRI aussi. On a publié un papier, il fait que 3 ans, où on a travaillé sur ça à La Réunion. La Réunion, c'est l'endroit parfait pour les réseaux, pour les gradients d'altitude. 50 km, 3000, c'est parfait. On prenait la voiture depuis le labo et en 5 minutes, on était à 2000 mètres. Et l'idée de la thèse de l'IRI, ce n'était pas de faire des réseaux d'interaction, c'était simplement de faire des études de diversité au long des gradients d'altitude. D'accord? Mais on échantillonnait dans les flores et on notait quand même dans quelle flore il venait chaque population, chaque communauté. Et c'était là où Giacomo Morassuti est venu à faire son master ici. Il était censé de faire des manips au laboratoire, mais la Covid est arrivée, il était enfermé chez lui. C'est là où je lui ai filé ces données de NIRI pour explorer des questions que, même si le jeu de données n'était pas fait pour ça, pour un master, je me suis dit, bon, on va explorer ces idées. C'est là où Giacomo il a fait un réseau d'interaction fleurs trips, parce que ces trips-là, on les échantillonne dans les fleurs. On prend les fleurs, on les tape sur un plateau, tu vois un tapis de petits points noirs qui bougent, noir, marron, blanc, et on les aspire, on les met dans l'alcool. Et ça, on a construit ces réseaux, et ça, c'est les plantes, les fleurs, et ça, c'est les trips. Et la taille de la barre, ça représente l'abondance de cette espèce. Et en haut, pareil. Par exemple, on voit que Trips bourbonensis, ou bien celle-là, Trips réunionensis, c'est un Trips, une nouvelle espèce qu'on a décrite à La Réunion, ça s'appelle Trips réunionensis, qui se nourrit, on va dire, à 80% sur Dombea ficunea, qui c'est une espèce de plante endémique de La Réunion, et certains, ça nourrit sur Nuxia, qui c'est un autre endémique. Et c'est comme ça qu'on fait des réseaux. Et là, Et Giacomo, il a déjà trouvé des résultats plutôt intéressants. Il a trouvé qu'avec l'altitude, on avait une perte de diversité, mais pas de diversité, seule de diversité d'interaction. Combien de lignes on trouve entre ces deux niveaux, d'accord? Avec ces résultats plutôt encourageants, on s'est dit, avec Matryoshka, pourquoi on ne va pas un peu plus loin et on essaie d'explorer comment ces réseaux ils changent entre la flore, les trips et la couche symbiote. Bon, plutôt des symbiotes, des bactéries associées à ces insectes. On ne peut pas vraiment parler des symbiotes, parce qu'on ne sait pas qu'est-ce qu'ils font. Et ça, c'était un projet exploratoire d'UCMEP où on a démarré cette ligne de recherche avec beaucoup de collaborations à niveau du CVGP, surtout avec la plateforme Biomol, encore avec Anne, avec Bruno Michel pour l'identification de trips. Dans ces projets, il n'y a pas encore d'encadrement. C'est un projet exploratoire. J'espère que ça va ouvrir des portes pour l'encadrement futur et pour des appels d'offres dans les années à venir. Mais en tout cas, merci au plateau biologie moléculaire et plateau collection pour ces chouettes collaborations. Les hypothèses, ça c'est le réseau tritrophique. Hein. Ça c'est les fleurs, ça c'est les trips, ça c'est une espèce de trips qui est associé à tous ces micro-lignes ici que vous voyez. Ça c'est des espèces de bactéries associées à ces trips. Pour faire ces manips, on a pris chaque trips individuel, on a mis dans un puits et on a séquencé cet individu pour voir, parce que comme ça on a deux puits avec deux espèces, la même espèce de trips, on peut voir avec qui ils sont associés, faire un réseau. Sinon on ne pouvait pas le faire. Si on fait des métabarcoding, on ne pouvait pas faire un réseau. Ça devient un peu cher et beaucoup de travail. Et les résultats que je vais vous présenter aujourd'hui, c'est qu'un 20% de séquençage qu'on a fait jusqu'au moment. C'est pour ça que c'est un projet qui démarre maintenant. Bon, en tout cas, on hypothétise que les réseaux plantes-trips, avec l'altitude, il va y avoir plus de... Le réseau, ça va devenir plus généraliste, il va y avoir plus de connexions. Et ça, on va le mesurer avec un index, que c'est le Network Speciation Index. Il y a plein d'index, mais on a choisi celle-là. À niveau de les réseaux Trisbacteria, on n'a pas vraiment d'hypothèse. L'hypothèse, c'est qu'il va y avoir un effet d'altitude, mais on ne peut pas vraiment dire, parce que, dès ma connaissance, il n'y a pas eu de travail comme ça dans le passé. C'est là où je pense que c'est une ligne de recherche que je trouve intéressante quand même. Pour ces manips, on a établi 7,35 d'altitude au sud de la France, de 0 à 1600 mètres. On établissait un site chaque 300 mètres, 6 sites total. Et à chaque site, on cherchait toujours 9 plantes pour avoir vraiment la puissance de faire un réseau plantes-insectes. Et de chaque plante, on récupérait les trips et on les séquençait entre 5 et 10 trips par plante. La somme, ça fait un gros tas d'espèces à séquencer, c'est pour ça qu'on y va tranquillement. Et on a barcodé le barcode bactérien et l'insecte pour trouver les espèces d'insectes. Bon, ici, je vous montre les résultats préliminaires de ces réseaux tritrophiques entre 0 à 1500 mètres. On voit par exemple qu'à 600 mètres, on a un réseau flore trips pas très complexe, on n'a que 5 espèces de trips et en basse altitude, on a un peu plus de diversité. Chaque réseau il est construit à partir de 7 réplicats, que c'est les 7 transects qu'on a fait, et de chaque réplica, on obtient une valeur de Specialization Index. Et ça, c'est les seuls résultats que je vais vous montrer de ces volets. On a observé déjà qu'il y a une corrélation significative. Ça, c'est le réseau trips bactéries. D'accord? On voit qu'en haute altitude, les bactéries, les réseaux deviennent un peu plus généralistes. Ça veut dire qu'en haute altitude, les bactéries vont s'associer à un nombre supérieur de trips. C'est un peu bizarre parce que l'index c'est spécialisation. Ça veut dire que ça c'est l'index spécialisation, mais c'est en bas qu'on a les généralistes. C'est un peu pas très intuitif. Par rapport à le réseau plantes trips, ces réseaux-là, on n'a pas encore assez séquencé pour avoir suffisamment de données pour faire une corrélation. Parce que chaque pointe, c'est un site. Et pour avoir des données de chaque site, il faut vraiment séquencer toutes les plantes. Et des fois, tu ne peux pas faire un réseau avec 2 plantes et 5 trips. Bon, pour conclure de ces parties-là, les trips, je trouve que c'est un bon modèle d'étude pour les réseaux d'interaction. Le travail de Niri La Réunion, on a chopé 5000 individus en 2 semaines de mission, avec pas mal de diversité. Évidemment, ce n'est pas la diversité qu'on peut trouver au niveau des coléoptères, hors question. C'est un bon modèle pour les études de symbiose chez les insectes. Et une chose qu'on aimerait explorer, c'est est-ce que les réseaux flore trips et flore bactéries, ils vont changer de manière différente si c'est un réseau plus mutualiste, plus généraliste. Ça, c'est une chose qu'on peut discuter après, peut-être en détail. Et une question importante, c'est qu'on n'a pas une bonne base de référence moléculaire au niveau de l'identification de trips. On va la créer avec ces projets, que ça peut aider beaucoup les agriculteurs à avoir un séquençage de trips et savoir quelle espèce il attaque les cultures, par exemple. Bon, là, je suis proche de la fin. On est à 37 minutes. Alors, c'est 10 minutes de plus et on est bon. Je vais faire un petit groupe de conclusions sur pourquoi on est intéressé à l'écologie communautaire, un peu sur le mentorat avec les étudiants et les jeunes chercheurs en général. Bon, je pense que mon approche à l'écologie communautaire, c'était toujours, le départ, c'était toujours ma thèse, un ravageur qui attaquait une plante, parce que le biocontrôle, la lutte biologique, c'était un point central de toutes mes recherches. Mais si on ne comprend pas toute cette complexité, on ne va jamais réussir à contrôler un insecte. Et la plupart des échecs de biocontrôle, c'est à cause de ces manques de connaissances. Par exemple, les années précédentes, au début du siècle, on lâchait des ennemis naturels pour contrôler une cible. Et cet ennemi naturel, il décidait d'attaquer cette espèce qui était une espèce indigène et on avait une catastrophe. Je pense que l'écologie communautaire, ça peut nous aider de pas mal d'éléments sur ces niveaux-là. Je pense que c'est aussi important l'écologie communautaire pour attaquer un peu, pour comprendre un peu la crise de diversité. On a des données vraiment puissantes pendant les dernières années de la perte de biomasse d'insectes qu'on a même dans des écosystèmes qui nous étaient jamais touchés pour les personnes. On sait aussi que les extinctions, c'est pas une chose simple, hein. C'est pas que cette espèce disparaît parce qu'elle disparaît, elle peut disparaître parce que cette espèce-là, il a disparu et ça fait un effet cascade qui nous va arriver à nous. Bon, il est en train de nous arriver à nous déjà. On a publié un papier récemment avec Dirk et Rachel qui a attiré beaucoup d'attention, où on parle un peu de ça, comprendre les extensions en cascade pour comprendre bien les déclins des insectes. Vous voyez ici 2 réseaux trophiques. Bon, c'est le même réseau trophique. Ça, on va dire que ça, c'est des plantes et ça, c'est des pollinisateurs. Ces plantes, ils dépendent de ces pollinisateurs pour survivre. Si on élimine ces pollinisateurs, pas grand-chose parce que cette plante, elle peut s'associer à les autres. Mais si on élimine celle-là, on va avoir une extension en cascade de ces 3 plantes qui n'ont pas de partenaire. C'est là où l'écologie communautaire peut nous donner beaucoup d'éléments Dans ces papiers, on parle par exemple de que les herbicides, les insecticides et même les antiparasitaires, ils vont s'accumuler dans les réseaux trophiques, ils vont arriver dans les réseaux trophiques de plantes et ça va déclencher des extensions en masse à niveau des pollinisateurs. Bon, ça vient de la part un peu plus compliquée pour moi, parce que je suis un peu moins habitué à parler des questions éthiques en public, mais bon, je ne sais pas si en HDR, c'est un bon moment pour le faire. Moi, j'ai suivi beaucoup Jen Hemstra, qui a lancé une initiative qui s'appelle Mentor First. J'aime bien ces phrases-là qui dit on ne peut pas avoir une carrière en académie sans avoir un bon programme de mentorat. Il faut se préoccuper pour les gens avec qui on travaille. Il n'y a pas de carrière si on ne le fait pas. Et elle dit que si on va des bons programmes de mentorat, ça va donner plus de créativité et plus de productivité dans les laboratoires. Qu'est-ce qu'on peut faire à niveau pratique? Bon, toujours cultiver un environnement inclusif où tout le monde se sent bien. Prioriser le développement personnel des gens avec qui on travaille, ce n'est pas toujours facile si on a un étudiant qui veut faire une chose qui ne nous arrange pas pour notre projet, mais ça devrait être la priorité. Être transparent, pas qu'avec les étudiants, mais aussi avec les partenaires des projets. Évidemment, promouvoir le bien-être dans les laboratoires. Une autre chose que je voulais parler, c'est de l'Open Access Publishing. Je pense que c'était une révolution les dernières 5 ans. Ici à Montpellier, c'est un pôle important de cette petite révolution. Ils ont créé cette système cette database où on propose des journaux pour publier qui sont associés à des sociétés savantes et pas que pour le bénéfice, le profit économique. Il y a l'initiative Peer Community très intéressant où on a des PDF qu'on va recommander où les auteurs ils peuvent publier sans payer et les lecteurs ils peuvent lire sans payer et ça va nous faire tous gagner. Et finalement des questions de genre, je ne vais pas vous faire un discours sur la parité en académie mais je voulais partager une expérience personnelle. J'ai pris le temps de faire ces graphes qui c'est les barres c'est les années depuis la première publication que j'ai faite. Et en vertical, on a la proportion de hommes et femmes avec qui j'ai coautoré des papiers. Et on voit en rose les hommes et en bleu les femmes. Et on voit qu'au début de ma carrière, j'avais 3 directeurs de thèse hommes. Mon premier PI, c'était un homme. Mon deuxième PI, c'était un homme. Ma carrière, au début, c'était 100% des hommes. Quand j'étais embauché au CIRAD 2016-2017, j'ai remarqué ça. Et j'ai fait un peu d'attention quand on a des invitations pour écrire des papiers, tout ça. Et je pense que ça a été clé pour avoir ces petits changements qu'on voit qu'aujourd'hui, même s'il y a des mauvaises années, aux différentes années, mais là aujourd'hui, dans les 2 dernières années, on était proche de la parité. Or, les discours comme ça, je pense que la chose que je voulais partager ici, c'est pas toujours facile de mettre en place un bon programme de mentorat. C'est pas toujours facile de publier dans les journaux. On pourrait publier toujours qu'à PCI, mais je pense que l'important c'est d'être réaliser qu'on a des biais très importants et que le fait de penser ça peut faire un petit changement. Avec ça, je voulais finir avec un grand merci à beaucoup de personnes ici au labo, dans mes postdocs et au passé. Et finalement, un grand merci à toutes les personnes ici et au chat de votre attention. Merci beaucoup. Merci.
Si vous voulez sortir, c'est le moment.
Bien sûr.
Ouais. À toi on peut arriver là. Je pense que vous allez rester seul J'avais pas allumé, arrêté mon téléphone.
Ça réveille un peu. Ouais, c'est bien. Faut pas laisser ouvert. Si je vais se garder très.
C'est parfait, non? Bon, donc on va faire Elodie, Elisa, Fabrice, Christophe, Alison et moi en dernier. Donc on commence avec Elodie. C'est parti.
Ok, donc on fait les questions en anglais, c'est ce qu'on avait dit tout à l'heure.
Yes, please.
First, thank you very much for the invitation to sit in your jury. I really liked reading your document, especially because the topics you work on are quite far from what I'm doing, so I learned a lot, and I thought the document was very well written, and I just loved the pieces of art. It's your cousin that made them?
Yeah.
It's amazing.
She made it quickly, yeah? She's very good.
That's beautiful. It makes me think that probably every institution would benefit from having scientific illustrators because it gives something more to the research. Okay.
She's always looking for a job.
I wish I could. So thanks a lot for that. And the amount of the work you're describing is super impressive. Like, I don't know, maybe you sleep very little hours. It's a lot of work. So
thank you.
Congratulations for that. And I will have just a few questions really on the science and a few more general ones. On your intro, you tackle the issue of the diversity-control relationship, and you focus mostly on complementarity to explain this relation. And is it like the consensus? Because I had the idea that probably some other effects like insurance effects and maybe the evolution dynamics of resistance. If you have more enemies, you're less likely to evolve resistance because you should evolve multiple resistance at the same time. So,
that's a good question. Well, I think that the key question that rises all the time with this kind of studies is the complementarity. I think that it's, I would say, the main mechanism. But then, of course, there are the thing I can think more based on my system would be basically behavioral interactions, for example, where they can one escape on the other. But apart from that, maybe I'm missing something here. Of course, there is always the sampling effect, where you just you do the experiment specifically to see this kind of interaction, and you are missing a lot. But I have a hard time to think about other mechanisms. Am I missing something here? Maybe.
Yeah, I was thinking
probably,
that maybe, especially with insects, you have a lot of unstable dynamics between boom and bust dynamics. Sometimes, you need another one to cope for the one that is done at the same time.
Yeah, for me that would be kind of a complementarity too, because it would be kind of a temporal complementarity. In fact, the first experiments for the project that I'm developing now is exactly this. We put a wasp that overexploits the aphid, but then we put an intrahil predator, a ladybird, that will eat some of these wasps that will prevent this burst. And it doesn't work for the moment. But in that case, could we talk about complementarity? I would say it's complementarity. Even if one attacks early in the season, the other by the end of the season, for me that's complementarity. But then we can fraction this complementarity into different steps.
Okay. And I was interested with meta-analysis that you're showing, the Danish et al. 2019. I didn't know it. And it shows a super strong relationship between diversity and control. And I was curious because people who have been working on biological control, conservation biological control, they've been struggling for like the last 15 years to show that it works. And mostly what they found, because they try to improve the diversity based on notably the floral diversity and the plant diversity with flower strips and whatever. And they find that it does improve the diversity of insects and natural enemies, but they can't make it up to the control. It doesn't work that way. So I was wondering if the Danish paper was based on non-manipulative data, like it's observational data only, and maybe communities that are diverse in nature just by themselves do work better for control, and when you try to improve them kind of artificially, you can't get the same.
As far as I remember, but I'm not 100% sure, I think that this paper is based on observational data in the field. I say that basically because all these data points, I don't think there's enough papers to make this such a large number of dots in the map. So I think that it's mostly observational data where they took, for example, by control services. The classic experiment for this interaction is you take your aphids, you put them in the field in different systems with more or less diversity of natural enemies, and then you measure suppression of these aphids. So I think this is the type of data. But it's a paper that I've been using this slide for 3 years, and now I don't remember exactly the paper, but I'm almost sure that was the case. But from this to have a flower stripe that is able to have enough spillover of enemies to your field, that's another story. I think that these are studies, these enemies, they disperse more than we think. And usually it's like at the landscape level where we can estimate this diversity of natural enemies. It's not like we put some flowers in between rows of tomatoes and it's going to be a miracle. It's more like at the landscape level, I would say.
Okay. I have some questions on the first part of your talk, the one with yellow and red mites.
Yes.
And I was curious because could you go back to the slides where we show the complementarity effect? Yeah,
that one.
So next one, the one where you have the bar plots.
The bar plots, yeah.
Okay, so this one.
Can you see it?
In this one, when you have both species of mites, you do manage to suppress the red strips better.
Yeah.
And in the next slide, you say that the red might disappear when they're both together. So how is it compatible with this hypothesis?
Yeah. I didn't put the dynamics here because all this data is a really compacted amount of data that comes from a dynamics. And what I think happened, but we need to analyze the data more in detail, is that at the beginning, these two mites managed to coexist pendant 7 weeks, and each one thing which is very interesting is that this predatory mite, it disperses in a very chaotic way. We even put some little wells in the cages, and we recovered 90% of the mites that we recovered were this species. So one of the hypotheses about the survival of this species in ecosystem is that it disperses in a kind of a crazy way without any kind of purpose, and by chance it can detect some population, some small patches of the prey. And I think that during these 7 weeks, this had an effect that then lagged behind from the rest of the experiment. In fact, if you see the dynamics, you see that in this specific treatment, you see there's a drop at the beginning and then they go up again. So the dynamics is not very beautiful. This is all why I didn't show it, because they are quite overlapping. It's not like this dynamics that you see very separated lines. It's at the beginning, and I think that we will need to analyze this by different time frames, because there's a beginning where they just really complement, but at some point, and that's the other thing, at some point the cages became so full of enemies and thrips that then this is when competition starts. I always say that in these dynamics, we see the real thing after 7 weeks, when the cage is full of insects and they start struggling for survival. And this is kind of struggling for survival is when there's not enough resources, when things are getting very tough. At the beginning you put 10 aphids, then... So that's my explanation.
And I was wondering if it could be... you wanted to say something?
It's just on that, but you can go ahead.
Yeah, it's the same idea. I was wondering if maybe you could have the red mite acting like an alternative resource to sustain a larger population of the good predatory mite, and so it would increase predatory pressure.
That's a good point, because we know that the yellow one feeds on the eggs of the red one. Yeah, that's a good. I don't think we have data to test for this. Well, we could do a kind of a correlation within the cage to see if there's a.
You can, if you have the data on the number of yellow ones, you can see if you have higher population sizes when they can feed on the red ones.
Yeah. Yeah, that's something that we could test. I mean, as I said, this is unpublished. It's good that we discuss about this because it can help us to just get this paper out, which has been in my desk for 4 years.
Yeah, maybe a question, because it was really on that side. So maybe a very basic question on the limitation of the system, because you show here that and you told us that the dispersion abilities of the different species are very different, and so maybe the containment in the cage is really a problem for the yellow one and not for the red one.
Yeah.
So how do you think the thing could be very different in greenhouses or open fields? And the next question is, I was almost a little bit afraid that this experiment led to a complete change also in the production of this biocontrol agent, because it's based on these very specific conditions.
Yeah. Well, there's different things here. Huh? The critique about the cages, and I think Christophe will agree, it always comes when you write a paper. It's like, oh, but the parasites cannot disperse. It is a limitation. Yeah. If you manage to find stability, meaning that all the species persist in this cage, it means that your result is extremely strong. Of course, the ideal would be to have mesocosms, larger, or cages that you can connect. But then at the technical level, it becomes very complicated. And the decision about not using this mite is not only from this experiment. We also did samplings in the greenhouses. This mite, we were doing some preliminary tests, releasing these enemies, and the red mite was just simply falling from the plants. So it was really. And another thing is that the yellow one is a spider mite that is used in all Europe to control thrips, and it was introduced in Reunion, we don't know by whom, and it's become like extremely efficient. But the Biofabric wanted to use the red one because it was local, there's no registration, so they could do everything that could be their own product. But I was telling them, I'm sorry, but it's not doing the job, so just forget about it. And they did.
Yeah, I was wondering also on the same line, because you say that after 7 weeks, you reach very high densities both of pests and predators, and so it unstabilizes your system. It makes one of the predators go extinct, and very high densities, like they're not natural, right? I mean, in a greenhouse, you will never reach that kind of density, or is it representative of what you can get?
It is kind of representative. I mean, these cucumber greenhouses, cucumber has the leaves like that big. They were totally covered with thrips. The flowers, you would tap on them, and it was like a cloud of little brownish dots, and they were defoliating strongly in some cases. Then in the sweet peppers was a bit different, because just very low densities, they damage the fruit, and then the fruit is totally very ugly, so you cannot sell it. But in some situations we have seen really huge densities. But I agree, it's a bit forcing the situation, but I like forcing the situation because this is where you are struggling to survive. Like, this is where you really have to be extremely competitive to manage to persist.
Yeah, but it does force an overlap of the niches that might not happen in nature.
Yeah. No, that's totally true. I mean, I think that in nature they just move away from the greenhouse and they hide in. And maybe this red mite. What I think is that this red mite is in the greenhouses because it comes from outside, it colonizes, it manages to stay there, and then it goes out. because it's an endemic, it's a tropical spider mite, endemic from the tropical region.
I had an interrogation. It's not really a question. Maybe you just can explain it to me. How do you analyze the temporal dynamics of data? I was under the impression that you were looking mostly at the final state of your system.
Yeah.
And do you plan on looking at more transitory dynamics? Is it relevant for the questions you ask? Because usually it's difficult because you've been looking at such long time series, it's so many data, and sometimes you just take the last one. It's so frustrating.
Yeah. I've been struggling with this question for 10 years now because I'm not a modeler, and I think that modeling can bring a lot to it. This is why in this project there's a modeler. What I do, I use R packages that are well developed for this. I've been changing packages every year because there's a new package. There's autocorrelation included. But there's no temporal correlation. There's only spatial autocorrelation. Then you send an email to the person. Well, it's difficult if the person that developed the package has a problem with that. So now there's a package that I'm kind of confident. What I do usually, I run with different packages. If I get the same result, I'm kind of okay. And one key element which I like a lot is the survival analysis with the extinction curves. This is non-parametric. There's an extinction that you see. I didn't present this data because I wanted to do something that everybody could follow. The survival analysis is a bit more complicated. But this is why I really like to do the survival analysis, because when you see that one species goes to zero and the other stays in different modalities, then you can really talk about coexistence. So this is my approach, with a lot of limitations, of course.
How much time do you have?
One last question.
No! I have so many! Okay.
We can keep talking next week. I enjoy this discussion.
Probably. Yeah, I was wondering about your approach to responsibility, both towards your students that you're mentoring, the society in general, on the question of pesticide and sustainable agriculture. And it's not a trick question, it's a real question. You do have a lot of international collaboration. You do attend a lot of conferences. And how do you conciliate that with the general idea of being more sober in our mobilities, in our consumption of energy, and like the cost, the energetical cost of science? Yeah.
Well, I mean, this is also something that has changed in the last few years. I put a slide at the beginning and then I deleted it about the impact we saw when I started my thesis. I saw this wall that we are going towards. Now this wall is like here, and it's really scary. So what I do is I limit the numbers of conferences that I take a plane per year. In 2 years, I have now a project with quite some money. There's this entomology conference in Japan, which I would love to go, and then take some holidays maybe after, and I'm not going there, because I thought it was too much. Maybe I will go one day, but this year I decided not to go, and I think about it twice. Again, you do not act always in agreement with your principles. In fact, most of the time you don't. But just this little thing. But I'm quite pessimistic about what we can do in Europe. I mean, honestly, yeah. But this is.
Elisa? Okay, yeah, it's my turn. Well, thanks again also for inviting me for your jury. I really enjoyed also the report, and I learned a lot of things about interactions between aphids, parasitoids, plants. It's, yeah, really interesting. So in fact, I had also a question about complementarity. But I guess it's also because I know more of the literature on pollination for some reason. So in your project, you aim at measuring complementarity or estimating in a meta-analysis from trait data.
Yeah.
And I was wondering which type of trait, because not all traits might be related with complementarity.
Yeah.
So, yeah, what are your thoughts on that? And
yeah. As I always say, sometimes you put things in a project that read very well. When you want to do them, it's another story. We are starting this line of research with Francisco, with a PhD. And the key element is to find some, it's true that, for example, the size, you might think it's not relevant for biocontrol, but I do think it's relevant. So basically, if we have, for example, with the spider mite system, we have these predatory mites, which are 0.5 millimeter, and then you have the predatory bugs, which are 1 centimeter. So just because of this, I think there's complementarity, because the little one has a 5 days cycle, and the other one needs 2 weeks. Maybe I take a vision of complementarity which is very general, and maybe I'm wrong. In fact, when I submitted the project, then I was reading papers like, oh, it's not clear in the project, maybe they will hammer me with that. But it's I present it in a very general way. I think that if you have a bug like this and a bug like that, they complement each other just by being different. So it's a functional trait diversity. This is how we will calculate this diversity.
No, but that's interesting because I was wondering, I know that for pollination, what they found for pollination of cultures, of crops, that for example, complementarity of pollinators were really related to like different timing in the day, even pollinating at different height, the flowers. And I was wondering if you had things like that also for parasitoids and natural enemies, or is it more related to life stages?
So this will be a tough part of the meta-analysis because we were working on the keywords to select the papers. We have around like 2,000 papers. I guess that we will extract data from max 400. But there will be species like biocontrol experiment in a country that is 2,000 kilometers from here using a native enemy, and finding the traits for this species will be very difficult. So I don't know how we will do that. I think the idea is quite interesting to kind of put like this complementarity as a measure related to the effect of biocontrol. And I think that size, that's something that we can easily find in then the life history generally, like are you a spider mite or are you a parasitic wasp or a ladybird? I think this is kind of in fact, in the project, the way we selected the complementarity communities was, or the non-complementarity was like 2 spider mites, 2 predatory mites, or 2 parasitoids. And I think it can be done, but it is not a perfect way to do that.
Yeah,
ideally, we will have enough data to measure all these traits. But,
thanks. So I guess I have to go fast because I have plenty of questions.
I will try to reply faster, sorry.
No, that's good to discuss. I have also, I guess, questions about the modeling part, because you're starting to include more models, community dynamics models. And I was wondering, well, first, a very general questions. What are your expectations from bringing your models in your experimental approach? Do you aim to more predict your experiments, like investigate particular mechanisms? What is your view on that?
Yeah. For in the project that we are starting now, there's a modeling that I forgot the name, which is a kind of a meta-correlation. It's kind of a path analysis where you can see how the densities of the different species correlate, and you can infer analysis. Okay, this you can infer dynamics. But from what I can more understand, which is something that we discussed already a little bit in the past, you and I, is that for example, we can, with a simple Lotka-Volterra model, we can just replicate this community, make it run, and parameterize it with the dynamics that we have, for example, an isolation of some of these species, that we have some papers that can give us the population growth potential, for example, and then say what happens if instead of having 1 generalist, we have 2 generalists, that this one also attacks this. What if this generalist attacks only these 2 and the other these 2? And then we can propose all these scenarios and then check for how many species coexist by the end of the day.
Okay.
But this is something that I'm still starting and learning. But maybe there's.
So it's more manipulating, like like the type of network that you have and see how your results can hold in different contexts. That's how you see it.
That's the way I see it.
Okay.
But of course, the interaction with the modelers, that was my main interest. I've been thinking so much about models, but sometimes it's very difficult to really see what is possible, that I want to collaborate with modelers to be able to know what is possible.
So, parameterizing, I always found it very hard, but I guess in a way models also can be a tool to think of different scenarios and different questions, because the results might depend a lot on the parameters you're not able to actually estimate. But
I was playing a little bit with the Lot-Volterra models in R, and it was like, if I change the initial number of aphids, it goes like... So I was like, okay, I need someone who knows what he or she is doing.
And I think it brings me to the next question related to the model. I know that in some cases of host parasitoids, you have estimations of, how many of the strength of the links in your networks. Well, I guess in many of the cases for your networks here, you can't estimate precisely the strength of the links.
Yeah.
Is it so? Okay.
That's true. And this is something that with this data we cannot do because the project is over. But with the project that we are starting now, we can easily estimate this interaction strength by doing simple experiments of one individual with one parasitoid, one control. Then we can measure easily growth of the one prey with the different enemies. And this is something that once we do the big community experiments, if we need some extra data to parameterize the model, then we can do these experiments. Yeah, that's a plan, but it depends on how the PhD works and the experiments.
Because one thing I was, I know less literature on natural enemies and predation, but for intragrid predation, from what I know from the theory, is that the stabilizing effects depend completely on the strength of interactions.
Exactly.
All the McCann papers on this type of things. And so I suspect that you can have destabilizing effects in some cases and stabilizing effects in others. And so estimating the strength of the links will be really...
absolutely. And this is something that we are starting to do. I mean, with Molly, that I showed the data, and with Anne, which started experiments here, what we were testing is just making combinations of intraguild predators. And theory says that they can only coexist if the intraguild prey, in that case the parasitoid, that it's hammered by the ladybird, it has a superior exploitation of the host. So what we did, we put the aphids, we called biobes, we put the enemies, and what happens is that intraguild predator is always more efficient at exploiting the resource, which is the opposite. The ladybirds, they arrive, they feed on all the aphids, they start laying eggs like crazy, and then in 1 week you have the cage full of ladybird larvae, and they feed on everyone. The parasitoid, it lays the eggs and they need to develop, so it's much slower. And this is where the conditions for coexistence in this situation is not possible.
And in your cage, is it the case they don't coexist?
No.
No, they don't. Okay, so it's coherent with,
The preliminary, the preliminary result is that there's no coexistence. They are quite disappointing. It's been like 4 months doing these little experiments, and we even combined predatory bugs with ladybirds, which are the two of them could predate on each other. It would be more reciprocal intraguild. No coexistence, but the problem is that we don't have enough resource. The aphids, there's not enough aphids to keep the populations alive. So that's something we are working on now.
And I guess that's, well, not exactly, I still have time. I still have time. It's not totally related with the model, but that's one thing I found really interesting, the part on plant volatiles that affect the interactions between the herbivores. Well, I knew a bit, but not much on this topic. And what I wondered is that, because in models we generally consider competition for resources, and it affects quite a lot the dynamics of the network. But what I was wondering is that what matters the most in this system? Is it like competition for actual resource of the plant, or is it more indirect effects between your herbivores mediated by these volatiles? Because it has consequences on how you model the dynamics, and I guess, it could be very important.
Yeah. Of course, the people working on this would say that everything happens through the plant.
Yeah.
The lab I was in Wageningen, they were having like years and years of experiments with this kind of questions. And something that, for example, when we talk about apparent competition, these two different herbivores that interact with the shared enemy, and then the speech is always, because insects, they don't really compete for the plant because they are highly specialized and they interact through the natural enemies. Mm-hmm. But the people working on plant interactions, they will tell you the opposite. No, that what's happening in the plant is extremely important. How relevant it is, I think it is quite depending, it depends a lot on the system. I think that, for example, in tropical ecosystems, in a forest with trees, most of the interactions are driven by natural enemies, because the plant, the way of connecting two parts of the same tree is difficult. But for example, in the cabbage system, which is what I work in the Netherlands, the effects are really, really strong. You put a larvae there, and the community of insects that will colonize this plant is totally different, even the parasitoids. So I'd say it's quite system specific. Then, of course, the evolution of plant defenses, not the induced defenses, the ones that are triggered after insect attack, the ones that are present there, these are the key to understand why a salad has some aphids and a rose has rather aphids. But this is the constitutive, it's like the basic structure of arthropod communities. I don't know if I replied to your question.
Yeah, I think you did. What I was wondering is that also in an agriculture context, when you have one crop, they all compete, in theory for the same crop, but the crop is highly abundant, and I don't know, in fact I have no answer to this question. I was really wondering, in the case of a crop where you have few, maybe not that many enemies,
I understand.
maybe volatiles at some point are more important. I have no idea, I wondered about this.
About volatiles, there's been a lot of effort trying to use these volatiles to protect crops, even with transgenic plants that emit volatiles and so on. And there's more failures than success.
Okay, so it wouldn't,
Because, of course, you can make the plant a bit more resistant, but it is just a bit more resistant. It's not enough to to have an aphid to say, oh no, I prefer to die before eating. It's like, okay, I would have a little fewer babies, but still I will attack you. So, and I think the natural enemies, if we talk about aphids again, I think that if you release natural enemies, is a kind of a clear trophic cascade.
So we can follow on modeling competition for resource and don't bother too much, maybe.
Yeah.
And if I have time for last questions, I wanted to ask a few questions on networks. Because I also found that very interesting in your presentation. I like networks, so so I think it's a question I have also when I analyze networks, is which type of network metrics you use in relation to your question. It's always a problem. And so in one of your first parts, you analyzed number of interactions. And I wondered, did you thought of other types of measures like vulnerability or even apparent competition? You can sometimes estimate it, indirect effects. And
I knew this question was going to arrive. Especially for the thrips part where we use this H2 for the specialization. There are so many different metrics. And this is something that I still need to think a lot about. Because, for example, one good way for the thrips part of the networks with elevation, I think that the good way would be to test against some null models. That would be the best way to solve this kind of sampling effect. For example, small networks, they are not, the metrics, we cannot estimate them very
very, very well.
Well, H2 is pretty good when you have this problem of sampling, so
Yeah?
I
Okay.
wouldn't.
Okay.
Yeah.
In fact, I took H2 because I was reading the latest papers and I was quite happy about it. But about this data, one problem that we have is that we have 8 communities. So technically, we have only 8 replicates, right? Because each cage with this community is a cellular replicate. And this is 8 replicates, you cannot say much. If on top of that, we calculated connectance and specialization in these networks, and then there were many communities which had the same value. So then we were even losing more power. And with these prediction links, we started with 11 until 3, and it was a good way to have a nice gradient. It's quite arbitrary, I have to admit, but that was our way to give to this data a bit more of power and a bit nicer gradient, especially with the integrated prediction links, because the max is 3. So you have 3, 2, and 1. When you model this, it's a bit. If you put it as a continuous variable, most models they don't fit. So it was kind of of course you can do a mean, then you do an ANOVA, but it's like, come on, we've been counting 20 weeks for just plotting it all together. Like, I cannot accept that.
Yeah. No, but I think it's always a question, and you have no choice with your network, but you have one species that have a lot more links than others, so you always wonder if it's just related to one species that just determines everything.
Yeah, I didn't present this part.
Okay, sorry.
I didn't present this part because what we analyze in this data set is, I didn't present that because I didn't want to make it complex. We did these 3 variables, which are predatory links, interpretation links, and presence of an absence of each of the enemies in the community.
Okay.
Like a binomial variable. And what we saw that was the most significant by a lot was the presence or absence of this predatory back.
Yeah,
When and we see it here, when this predatory back is present here, 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 and here, then you see these declines.
Yeah.
When it is not, with the spider mite is different because it prefers to feed on the competitors of the spider mite when it is present then. And this is the main way we want to tell these stories, like there's a keystone predation, there's one predator that plays a lot, but even with this strong influence, there's an influence of predation links, which is kind of interesting. That's
one last short one, if you have.
Yeah, it's maybe more a comment, but it's. I really liked also the network with symbionts and freaks.
Yeah.
It's different types of interactions. Yeah, it's great. And I was just wondering, did you think of using, multi-layer measures? I don't know if you've heard about this kind of.
I've heard of it.
Because I think one thing that might be great to test is whether, depending on the plant on which the thrip is, you might expect different symbionts. And I think with this type of multi-layer approach, you can maybe find if there are groups, of depending on the plant, you have different symbionts or not. But I don't know. Maybe.
Again, this is so preliminary. The analysis I did, they are so basic. I mean, I just made a correlation. But I think that this data, once it is all sequenced, it can be exploited for many, many years. Because you can test many things. We can even take the same species of thrips and see how many plants it fits with elevation, for example. That's a very simple thing to do. And I think that modeling in these 3 trophic networks, that would be something that that maybe we can discuss more. But yeah, there's plenty of things to do. And I think that the PhD student just to analyze this data would be great.
I think we're going to move on to Fabrice.
Okay, thank you. I will just continue on that a bit before congratulating you. But do you have the microbiome of the flowers?
No. But we can do it because it's a good excuse to go to the field.
Yeah, but this is it's probably something that's been needed to look at these things.
It is tough to know what these bacteria are doing.
Yeah.
Because maybe they are just bacteria that are there in the environment.
Yeah.
So this is why I don't talk about a network. It's an interaction network. They interact because it's there. But it's a great idea. We will do. Well, if you have money and time.
Okay, so well, first of all, I also want to congratulate you for the written report and the presentation. I completely share what was said by Elisa and Euproctis. It was a really nice moment of science, and not only science. And, I think the manuscript was very lively, and I think it shows well your talent for writing, and that's what we can see also with the opinion papers and the reviews. So it was very nice. And I also wanted to say that I think you have a lot of courage to embark in this kind of studies, because this has to be done, but it's so difficult. it's so fuzzy sometimes. So yeah, thank you for doing this, and also thank you for doing it like you do. I really wanted to say that I really like how you present your work, and also the humility and the ability to point out limitations, to point out problems, difficulties. I think it's something maybe a little bit new. I was in another HDR defense last week, and this was also said to the Matryoshka, who was defending. I think that it's very nice to see that the new generation of scientists are able to say that. It's something that I'm really optimistic when I see that. I think it's really nice.
I like doing these experiments, so it's not really. I'm still counting aphids, and I kind of like it.
Yeah.
I mean, it's especially when you plot them and you see the graph going to the right direction. It's like yes.
Yeah, but also saying that systems have limits, problems, difficulties. This is something that was not so easy to say before. I think it's changing. It's a new way of defining what is excellence, and I think that's very nice.
Thanks.
Okay, so I'm probably here to ask questions about symbionts, but I will first start with a question about your work on more community ecology.
Yeah.
And maybe in the beginning of your talk, you talk about observational studies and experimental studies.
Yeah.
And I was wondering, well, there is one big difference between the two also is that when you do observations, well, you hope to look at a system at equilibrium that embraces also all different things like variation in the environment, different genotypes for different species, etc. So, but when you do your experiments, you stop the system.
Yeah.
So it means that you have this initial dynamics. You hope that maybe you will reach an equilibrium. But maybe one question is how this dynamic, this equilibrium depends on the initial conditions and the environmental conditions.
Yeah.
And I don't know how to deal with that, but I wanted to know what you think about that.
I think the environmental conditions play a very clear role. In fact, for these experiments that we don't manage to get stability, one thing that we are thinking about is to put them just with 3 hours of light, so the aphids can grow and the natural enemies sleep. So we can give the aphids a head start.
Yeah.
Now this light pollution thing that is now like everybody works with the light pollution, it can be an option. About the initial conditions, I get this comment very often, and I always say the same. I don't think it's very important, even if modelers will tell me I'm wrong. But the way we do it is quite rough. We just put the plants, we put the aphids, we let them be in a good density, so almost to reach carrying capacity, and then we release the enemies. If the system is intrinsically stable, it works. And this is based on previous experiments with aphids. We had a paper with Alexander. We had these 3 aphids and these 3 parasitoids. You drop them in a cage. You come back 6 months after, while you put plants every week. And after 6 months, the 6 species are still there. The system is extremely stable. This is what I'm trying to find, and I don't manage. It's an exception.
Yeah.
It's an exception. Sadly, it is an exception. But this system was extremely stable.
Yeah.
And Dirk got rid of the parasitoids. I will blame him for that all his life. But if the system is intrinsically stable, the initial conditions is not that important. Maybe if the system can go to alternative states,
Yeah.
then, and we see that in the experiments. You start and then you see some cages going there, and the others like that. It's like, okay, these are 2 stable states. We do that sometimes with the final dynamics to put as a correlation in the survival how this correlates to the initial densities. We did that with Anne with this last experiment, and we found that it could have an influence, but there's a lot of variation too.
Mm-hmm.
So that's the problem.
Okay, well, I come to symbionts, and I hope that what I will say just now will not go out of this room.
Okay.
Because the first question I have.
We are live, huh?
is okay. Of course, symbionts are very nice because they bring very a lot of traits to their hosts.
Yeah.
But finally, if we put that in your work on community ecology,
yeah.
What does that change? That the trait is brought by symbionts or by an alternative genotype of the host?
Yeah, I always say the same thing. You do the same experiment. You have 2 genotypes of a PA fit, with and without Hamiltonian defensive symbiont. And one genotype is immune to this parasitoid and the other not. You do the same experiment, but what changes the susceptibility is the bacteria. And the journal, you can publish the paper, changes a lot. And I agree that when I was postdoc, I was driven by this interest in publishing good papers, because when I went for my first postdoc in England, I was there to do ecology, community ecology with aphids and parasitoids. That was the job that inspired me to go there. But all the lab was working with symbionts and getting really good papers, like, oh, come on, maybe this will help me to get a permanent job. So then I moved into this, and it works. But it's a bit unfair for the people, when we do experiments which are simpler. So I agree, and they bring, that said, the symbionts that Christophe works, I've never seen like a bacteria making an aphid totally immune to a parasitoid. I mean, it's like with intrinsic resistance, you can have a 20% reduction, but I had some Hamiltonian strains which were like, the aphid was totally immune to the parasite. And the fact that these symbionts are there means that there's a lot of selection for them to be there, and they're costly. Mm-hmm. So, but there's of course this kind of tendency.
Okay. So I will go on this question because, in my opinion, there might be situations where it's very different when symbionts and aphids do not have exactly the same interest.
Yeah.
Because finally here, the traits you are looking at are mostly mutualistic traits.
Yeah.
But of course, we know that there are also other symbionts that have selfish strategies.
Yeah.
And then maybe in that case, of course, adding the symbiont would be very different because, of course, this cannot be an extension and only an extension of the host genotype.
Yeah. You can simply add a costly symbiont that is providing a service that in your experimental condition doesn't matter.
But, like, this is almost what trade-offs are.
Yeah. You mean, for example, a case of Wolbachia that is manipulating sex ratio?
Manipulating behavior. For example, so it's not in the I will find the word, but for example, we did some work in Lyon on viruses that manipulate the behavior of parasitoids.
Yes.
And this is a selfish strategy of the virus, and in that case, it changes the ability and the competitive ability of different parasitoids. But there, it's something that is really independent on mutualistic interactions between the partners.
Yeah, I mean, I think that when you get into these kind of interactions, I tend to treat these symbionts in terms of intimate interaction, and simply as a parasites. They have their own interest, and of course, if they are quite specialized on the host, they want to get replicated to the next generation to make sure that there's no extinction of the host, because if not, finito. But then this is a kind of a struggle between different species, like parasites that manipulate hosts. And I would say that with symbionts like Wolbachia, I would say that's the case. It's a bacteria that is producing a selfish phenotype, and there's a stabilizing dynamics where if the selfishness becomes too extreme, then the whole system goes out. So that would be my view on the question. Of course, then what we know about this type of symbiosis is always very limited in the lab. When we check these interactions in the field, there's more and more papers coming out of these situations in the field. Then things change a lot, and the community context is when it comes into play.
Yeah. So, and maybe another question regarding this is okay. So you are also moving from almost strictly vertically transmitted symbionts.
Yes.
But also with gut symbionts, with the possibility that you acquire them from the environment.
Yeah.
I was wondering whether this is also something that could really change things, because traits can be acquired directly from the environment and be changing from one generation to the other.
Yeah.
And so it means that we maybe should include some kind of dynamic in the system, but then it could also change things on how the symbionts could impact the community.
The community. Yeah. As far as I know, for example, the gut symbiont and the surface symbionts and all these kind of symbionts, at the experimental level, but correct me if I'm wrong, at the experimental level we are still in a phase where we are describing. We are checking for funny phenotypes in the lab, funny and very interesting. But from this to get into a community context, a population dynamic context, we are still very far from that. And this is a little bit, this would be my kind of dream, to put like this experimental community approach and the symbiosis together, which is what we are starting to do with the thrips. We have some thrips in the rearing that maybe we'll sequence and try to manipulate, put them in cages, and see how we can move the field a little bit. But we are still very early into a describing phase, and I hope I'm not wrong, but as far as I know, there's not a lot of papers checking gut communities of butterflies, for example, in the field. They sample them, they describe them, but they don't do manipulative experiments. It is difficult, that's the other question. It's very difficult to do this kind of experiments, but maybe. Yeah.
And so regarding the description of the symbiont community in thrips.
Yes.
So, could you, if I understood correctly, you have more generalists, symbionts with elevation. Is that right?
Yes.
Am I right? Yeah.
This is elevation. This is the specialization index, so more generalist.
I was wondering whether you could correlate that with the diversity of symbionts you observed. So is it because you have less symbionts observed at high elevation, so that you also reduce competition between symbionts?
Yeah.
And you could get exactly the same thing. So what could be the driver actually of this kind of pattern?
Well, apparently the H2 index is quite robust to the difference in diversity. There's plenty of metrics, and what I liked about this result, which is extremely preliminary, is that I plotted all these different metrics that you can get from R, and there were a lot of significant effects. Of course, significant, bravo, but you can. This is why I selected the H2. But when you see these kind of patterns, you see that there's something happening. And well, as I've said, I would say that for a little bacteria here in low elevations with maybe with more diversity of plants, these bacteria travel with the thrips, and then there's this more diversity, and they can engage or can adapt it to more species. But in tough conditions, it's 1,600. It's not extremely tough. Right? It's not like 6,000. I would say that these bacteria, they just rely on more thrips to persist. But yes, there's a lot of conclusions here that we still need to think about. I risked a lot because presenting this is so new that I thought about it, but not enough. But I think it's nice to show things that is not they are not already reviewed in journals for 10 times. Well, the basic hypothesis is that you are a generalist, you can rely on more partners. It is a good strategy. If you rely on a single one, the extreme case, if the strip this year is not showing up in the particular flower that you were expecting, that's the end of the story. Then, of course, I think there's a lot of work in these little lines here to see which ones are potential mutualists. If we see Wolbachia, for example, clearly it's not an environmental bacteria. There's quite some Burkordelias also. There's some Hamiltonellas, there's a lot of Wolbachia. So maybe we can treat these different bacteria in a different way.
Yeah. May I quickly step in there? I think the interpretation of that pattern will depend on the result of the other pattern, because let's be honest, you don't know whether these bacteria need the thrips.
Yeah.
If these thrips at high elevation are sort of forced to use more diverse flowers, it's almost unavoidable that they will have more diverse bacterial communities that may simply pick up.
Yes.
Yeah, that's a good point. And of course, we need to subset a lot with the type of bacteria. But if we find the opposite pattern, that would be quite cool, because it means that we have a mutualistic network. And there's another question. Are thrips mutualistic on flowers? We don't know. They feed on pollen, they can fall on thieves. They can also transmit pollen. We don't know. We don't know because it's not a very well-known field, the thripsology. So, yeah.
One last question.
Okay, last question about mentoring.
Yes.
So I really like your the way you describe the way you mentor people. But we have discussions in the lab at the moment on what is PhD. So with people doing their PhD currently and older people. And one of the points that distinguishes PhDs nowadays and before is that before very few PhD projects were based on projects, funded projects, which is the case now.
Yeah.
And of course, this leads to maybe less freedom for students because, well, for example, if we look at your enemy cocktail project, everything is very clear, settled.
Yeah.
So how do we handle that? And how do we give conditions for creativity if the projects that the PhD students have to work on are so fixed?
Yeah. Difficult question. Well, the first thing is that when we select the candidates, we already mentioned that there's a project already. There's some minimum type of tasks that need to be developed, but of course there's a kind of a creativity on the side that we need to explore. In practical terms, the two previous PhD that I supervised, they were totally free because they had like really open projects. Of course, I was there to show what I think it was interesting for them to get the papers and so on. They managed to get the both of them. They were originally from African countries. They managed to get jobs in France, so that's their objective in private sector. But they got so that's my main objective. What do you want to do after your PhD? You want to get good papers and get a postdoc? You have to do this, or I think you have to do this. If you start doing things that are really not interesting, maybe you will not get enough papers to get this point. For the current project, it's 2 months. I don't know how it will go. I can imagine a scenario where this candidate says, listen, this project, I find it extremely boring, I don't want to do that, I think I would accept it. Even if I wouldn't be happy about it, but I would accept it, and you do something that it's in the lines of our research. Imagine that, for example, this person says, I want to do more behavioral stuff instead of counting. You have to follow, but it's then I would accept it, and then I would do the experiments myself. I mean, what else? Or try to get money to do the experiments. I mean, as I said, there is no way you can force a person to do an experiment. A student, a PhD, a master, a technician, there's no way. You don't want people working on something they don't like. So, and then you just get frustration out of the project, but that's how it is. What can you do? I mean, that's my view.
That's a very complex question. I don't have a clear answer.
I think that in that case I would accept it, even if I would make clear the point that, listen, this is not why you were here, but I accepted you changed your mind. Yeah, and I will cry.
Okay, thank you.
We need to move on to Christophe.
It's my turn. Well, thanks first for this very nice talk and for inviting me to beautiful Montpellier. I came from the snow, I ended up in the rain. And thanks also for crafting a really nice document. I mean, since we work on, or at least partially work on very similar topics, I have, of course, followed your work closely, and I was always impressed not just by the quality of your research, but also by its presentation. You're clearly someone who kind of takes pride in not just writing down what you find, but writing it beautifully, making papers easy on the eyes, and this really shows in this document. It was really a pleasure to read. You write a lot about biological control in your thesis, but if you had written that you did all this work to save the world's crops, I don't think I would have believed you. You didn't write that, but in a way I guess that's the beauty of working on multitrophic interactions centering on insect herbivores, that it's almost unavoidable to hit on findings that are clearly relevant in the context of biological control, and that can maybe be used to improve plant protection. But then you can do two things, right? You can say, okay, here's what I found, please somebody pick up the ball, I move on to the next interesting question. And you decided to go the other way, to actually now develop a project, and congratulations on getting it funded, to actually try to really build, let's say, mixtures of natural enemies that should work better than the best of them alone. Yeah. So I agree with Fabrice, this is a courageous thing to do. So I'll first talk a bit about biological control, I guess. You write one of the advantages is that it minimizes the risk of resistance emergence. Do you really think that's true?
Wow. Did I write it down myself?
Ça minimise le risque d'émergence de résistance.
I was a bit tired that day. No. Well, I guess that this is the classical sentence that we say that with insecticides, on top of polluting environments, we have resistance. But it is true that there's resistance against natural enemies. But I think that one of the advantages is that even if we have resistance, we have a lot of diversity on the other side to counter this resistance. Of course, we have plenty of chemicals that we can produce, but most of them are now forbidden for good. So I think that was a slide that I deleted where I wanted to say that we don't need more solutions, more technicity about biocontrol. The diversity is there, we just need to use it. So I think that there will be resistance, but we can counter this resistance with all the diversity we have at the natural animal level.
I think it's probably we have more tools to avoid it in biological control, but I think it needs a conscious effort actually of the producers to, I'm not sure the currently used commercially available natural enemies are that much more diverse than
this is also true. This is also true. But our knowledge can make it maybe. In our cages, there's no way they resist the reservoir resistance. I would like to see that anyway.
Then you also talk about a lot about the biodiversity biocontrol relationship and the complementarity that we already touched upon. I think this complementarity of natural enemies could be particularly important when you have resistance conferring symbionts in your pest populations. Could you maybe elaborate on that a bit?
Yes, I can try. Yeah, that's a good point. It's true that the way we presented this data, there was no resistance involved. It was very dry. We do these cocktails, we put them in cages, and let's see what happens. Obviously, with an hypothesis behind. But it is true that this complementarity, it can be the way of using multiple enemies that can balance this resistance and have this kind of this dominant genotype which is resistant. It can be suppressed by one enemy, and then the other, they can be this balancing selection of these different genotypes. That's a little bit what you're asking, right?
Yeah, and also I think the symbiont conferred resistance is often sort of specific to particular types of enemies.
Yeah,
right?
Okay. Yeah, of course. And this, you recently published a paper on something similar where you have these different parasitoids that manage to maintain the diversity at the level of the symbionts, right? So, okay, I'll try to think a moment, because I think I'm talking a bit without thinking, and that's not a very good idea. So,
no, I mean, that is a very valid point. Not all parasitoids, for example, might be equally effective, but I also think about predators, right?
Yeah, of course, yeah. Of course, if you have a cage with a ladybird that is never going to be the symbionts is unlikely to protect against it, then it's a good way to complement it. Yeah, of course.
I think that would really improve some strategies. Also, the
generation
are quite, especially if you don't go in with high densities of ladybirds like you described.
Yeah.
They're voracious, but they're also slow, whereas the parasitoids are quite fast.
Yeah. Then another issue that I see with this kind of complementarity to counter resistance is that when the resistant phenotype emerges, for example, against the wasp, I think it is quite likely that this wasp that is not any more efficient any longer, it is likely that it disappears from the ecosystem. And then we end up with a system where we have lost one of the main players, and the complementarity is lost. So I think that one key element is how we manage to maintain all these species that at some point they are not useful because the host is resistant in this applied environment.
And that brings me to the next question, which I found it very interesting that you said, well, sometimes intraguild predation might not be a bad thing. It might be a good thing because it kind of allows for longer-term persistence of multiple metroids.
Yeah.
I mean, I guess in this sort of mass rear and release type of biocontrol, I guess long-term stability is not always the goal. You just want to kill your pest before you can harvest your crop.
Yeah.
But in other forms, it is important. And I think that's where the modeling could be important, because I think it might just act via the growth rates.
Yeah.
I remember teaching a lecture to first-year ecology students, taking a normal logistic growth model with discrete generations, and then you go from a low R to a high R, and you go from beautiful sigmoid approach to K to overshooting to 1 phase cycles, 2 phase cycles, and you end up with chaos with the very same equation.
Yeah.
And the things you're working with here, thrips, aphids, they have enormous growth rates.
Yeah.
You can't see them breeding like rabbits because rabbits are much slower.
Yeah. No, that's, they grow like bacteria almost.
Really, and I think that is part of the reason why it's so hard to find stable communities like the one Deer kicked upon. And so it might just be somewhat reducing the R's through intraguild predation that might already lead to stability or better stability, but I guess this would need a kind of modeling approach to verify.
Yeah, I think that the data that we have already, they are enough. It's powerful enough to put some models on it and to see why it is failing. Then maybe look for another type of interaction enemies. The advantage is that, well, two different things. One is that with the partner, with BioBest, they have so many enemies that it's very easy to play around with them. Of course, to test the basic hypothesis of the paper, of the project, maybe that's a bit more difficult. In fact, when I was proposing the combinations already in the project, I was like, these two, it's not going to work, it's impossible. But in paper, it reads well, anyway. But it's true that the reality is quite different. What I was going to say about equilibrium in biocontrol, it is true that if we manage to get a cocktail that is very efficient, that can suppress populations, and that's the end of the story, maybe that's a good strategy from the company point of view, because then they can sell over and over. But if we manage, ideally, to get a cocktail that maintains the populations below the economic threshold, which are always present there, when there's a new aphid arriving, it will be hammered because the enemies are there, that would be quite beneficial also for the company. Of course, below the economic threshold. And we know it's not likely.
It's not beneficial for the company because they sell it once and they can't resell it.
No, but if they sell it once a year to many greenhouses, and it's like that's the stable, the long-term, maybe some farmers cannot afford releasing these enemies every month. So, yeah.
And that is again a nice connection to the last question on biocontrol. I'm actually quite optimistic that biocontrol, in a way, it has almost become a bit the method of choice, right? So some go completely with natural enemies, others do the integrated production. They start with natural enemies, follow, the best population, and only intervene with insecticides if they need to. But if we really want to have an impact on, the amount of pesticides out there, we actually have to bring it to the field. And in my opinion, that hasn't succeeded yet. So what do you think is needed to...
You mean in open fields, eh? Not
basically go from protected crops to open crops.
Yeah, I mean, again, I think that the project that I just started, it's a good way to do fundamental community ecology with an applied interest, applied to a very limited condition, which is the greenhouses. And maybe I shouldn't say that, for me the production in greenhouses is not the best, ecologically speaking, because it's a lot of plastic, in some latitudes, a lot of energy. So ideally, in an ideal world, we would plant our lettuce in the backyard. So, to go back to the question about that you proposed, for the open field, I think that inundative biocontrol in greenhouses, it can be extremely useful. But in open fields, what we need to do is manipulative biocontrol, where we manipulate landscapes to bring in the natural enemies. And this is, for me, is the best way to produce crops. It's cleaner, it's more beautiful, it's affordable to everyone. We don't need bio fabrics that produce them. But as we are heading now, we can be happy about it or not, as we're heading now, is the production in greenhouses is becoming every time more and more important, especially with global changes, where some crops will not be able to survive outside these greenhouses. In Reunion, for example, thanks to greenhouses, we protected some crops from cyclones. So I think that my niche is there, but there's a big niche on this other biocontrol in natural conditions that is extremely exciting. And Christine works on that, and I think it's the way to go. But we need to find our niche, and I think I found it.
I totally agree. No. I think also in terms of yield per area, that becomes a limiting factor as well.
Yeah, but sometimes they put the plants into plastic, so that's not extremely natural. But
we have time.
Maybe some more specific questions that just raised my curiosity. Your beautiful paper on the Haemotonella infected aphids basically suppressing the plant's volatile alarm call. It was actually not just Haemotonella, that was almost all symbionts that triggered this. I'm still wondering about how. Because most of these symbionts are kind of costly. Is it related to the vigor and fitness of the aphids or?
No, because we corrected for that.
Okay.
We corrected for that. We measured the amount of babies produced when we induced the plant. So for those who doesn't know the paper, we've demonstrated that the presence of symbiont changes the volatile profile and reduces the attraction of the parasitic wasps. So we corrected for that. So we were measuring the babies produced in these clip cages used to induce, and there was no correlation whatsoever. So that was the same. This is a project that I finished when I finished my postdoc. I was expecting someone to follow up on this because it would be so easy to do experiments, to do metabolomics in the saliva, or even behavioral studies. So my guess is that there's something in the saliva that is going. We know that the saliva of aphids is changed when Hamiltonella is present. There's some papers showing that. I think there's some effectors that are filtered in the saliva that go to the plant and that block this response. Another thing that I think it's quite possible is that the behavior. We know that Hamiltonella changes the way the aphid penetrates the plant. These electropenetration graphs is something that you attach a cable to the head of the aphid, and then you know how many times it's doing this. And Hamiltonella changes these patterns. So maybe it's just a behavioral change. What is shocking is that it is doing that with all these different species. I was expecting Hamiltonella doing that, but not the others. And they all do that. So maybe it's the vigor that will be very boring, but we controlled for that.
Yeah. No, that explanation is much more interesting.
Yes. So there's symbionts also in the honeydew sometimes, that maybe they are deposited on the plant, and then when the aphid feeds on the plant, is injecting them in the plant. Hamiltonella is related to plant pathogens, and these plant pathogens, we know they block these induced responses.
Okay.
That's something that if I one day have the time and energy and money, I would like to go for it. But it's very functional, and maybe it's a bit far from what I am.
That would actually be an interesting explanation. Yeah, well, other questions are very detailed. Just one thing I wanted, your biodiversity related work. The transect you did on La Réunion.
Yes.
There were 106 flowering plants, of which 20 were indigenous. Is that the state of affairs in La Réunion?
Yes.
Is it really that bad?
Yes. It's
never been there.
No, it's a disaster. So like the island, there's a part in the east which is still quite well preserved, but the invasive plants is really the biomass. I remember when I was living there, we were doing like hiking around, and it was like, wow, 90% of the biomass is invasive. 90% most of the time, except if you go to this forest. In high altitudes, in low altitudes, it's 100% invasive. It's like, and there's a lot of losses of big birds that disperse these plants, that they limit. So there's some endemic trees that when this individual dies, end of story, because there's no dispersion. There's efforts to correct that, but yeah.
Thank you. Congratulations again.
Thank you.
Alison.
Okay, so yeah, thank you very much for asking me to be part of the jury. I really enjoyed reading your project and your talk today. And I'm looking forward to working more with you in the future. So, you probably guessed I'm quite interested in evolution, even though I am. So, what is the genetic variation in both the pest and the predator populations, or the interspecific variation? Because this could also have really strong effects on the effectiveness of biocontrol at all different levels.
Yeah, you're talking about the experiment where we had the
I'm talking about all of these experiments. So when you get your species from BioBest, how variable is the wasp? Also, how variable are the pest populations in each of you? I know in the aphids there's just one, they're not variable, but in the spider mites, for example.
Yeah, that's a good question. So this was a collaboration with La Coccinelle, which is the biofabric in Reunion. These rarings were kind of starting at that moment when we did experiments. For some species, they had some issues in terms of inbreeding depression, because the library was not working very well. That was our guess. So I would say that at the enemy level, the variation was quite limited. At the herbivore level, the thrips, we collected them from the field directly, because we couldn't manage to get them in rearing. And the spider mites was a rearing that we kept from the field, and that was relatively diverse. But to answer your question, I think that it would be very exciting to repeat these experiments from an eco-evolutionary point of view, where we put several clones of aphids, several spider mites, and see how this alters the dynamics.
That's definitely be really cool. But even aside from that, the genetic variation in the pest population may affect the ability of the predator to deal with it. Because if it's homogeneous, maybe it's like, okay, I just can eat it really easily. I can plastically adapt to eat this one. If they're a bit variable, maybe it's a bit harder because they have to upregulate different.
Yeah, it is possible. My guess is that what would happen in this kind of dynamics, imagine that we have several strains of this spider mite, and some are more or less resistant. I think that by the end of the experiment, the pressure is so high that we would have a strong selection for those strains that are less, more resistant to this enemy. So, but we never managed to measure that. That would be something that I would like to do, as I said. And the question is totally very interesting, and clearly knowing, for example, the diversity at the enemy level, that would be also extremely useful to see how they specialized on different resources.
Because the diversity at the predator level could affect the behaviour there and the intraspecific competition, because there's going to be competition for space, and then the result, which you said is weird, of that red mite,
that goes up,
eating its own eggs. Maybe that's to reduce levels of intraspecific competition and things like that. I mean, maybe it's not anything to do with that, but I'm just saying that the intraspecific competition could be really important as well for like the population crashes too. Because, what do you think? I don't know.
Yeah, I mean, these are questions that I think that the word variation at the level of all the different levels, and even evolutionary changes in all these different levels, is something that is extremely missing from my research program. And I think that we are missing a lot. We do it this way for practical terms. It's difficult to do otherwise. The fact that Propionopsis mexicanus feeds on the eggs of the competitor of its own, I don't think it could be a self-strategy to reduce competition, because what we did in this experiment, we offered them eggs of the two species, and then we released them for 24 hours, and then we saw the eggs that lasted in this experiment by removing the adult and allowing these eggs to develop into adults, and then we could count. So why eating on your own eggs when you have the eggs of the other? Well, you can argue that since...
But it could be to reduce, if I eat the eggs of the other, then my offspring will have more, survive more. They're reducing the intraspecific competition for the next generation, that kind of dynamic.
Yeah.
It may not be true.
Well, this is what one of our hypotheses, to eat the eggs of the competitor.
But the competitor species, not their own species, because intraspecific competition is really intense as well in these systems.
Yeah, okay. So you mean that, if I understood well, you mean that maybe by eating its own eggs, it can reduce the interspecific competition.
And it may respond
it is possible, but I find it quite a very stupid alternative, right? I mean, why would you kill your own kin?
But it's reduce kind and kin, because you might kill.
To ensure
if it's your sister's egg, you might not eat it. But if it's your
prefer just you think about your offspring and not your cousins. That's basically it. Okay, maybe it's possible, but I would say more like it's true that we don't have selection is not acting on species level, but on individual level. Okay, that's yeah. Why not? It is possible. It is possible. So what you are saying, it's very interesting because the way I solve my story is that this guy prefers to eat on the competitor to do better, but it makes no sense by the end of the day, because what you want is to think about your own offspring, right?
Kind of. Okay. I suppose what I'm kind of saying as well is that the genetic diversity of the predator may change the outcome of what you're getting in the cage experiments, because they might behave differently to kin. Related individuals, unrelated individuals is what
I totally agree.
Getting at.
Okay, so that changes again the way I could think about this data. I don't know if we have data to explore this question. Maybe in the cages here, it could be possible to check, like in the dynamics, if there's a density dependent decrease. So meaning that the nice thing is that we have the dynamics. So here we could test if when this species becomes very dense in the dynamics, then it can become a resource, or if this one is very dense, if it can be eating on their own. Okay. That's very interesting. That's something that maybe that's the beauty of dynamics, that we can test what happens at different densities.
They're cool. They're really cool experiments.
Okay.
And I don't know if you asked a quite similar question when I popped out, but when you have like the intra-guild relationships, complementarity with parasitoids, to me it's quite obvious because they can have a different species, or they can have or different life stage. But if it's like these mites again. How much do they actually compete for space on the plant as well?
The predatory mites, huh?
Yeah.
For space, I would say that they mostly compete for resource. I mean,
but they lay their eggs. Where do they lay that? They lay their eggs on the leaves as well.
Yes. But I mean, I don't think you can cover the whole leaves in eggs.
I don't have a clue.
No. I would say that the main competition is through the resource for prey. Then another interesting thing is that the spider mites, they do these kind of webs, and there's another predatory mite which is specific of spider mites, and this one is very well adapted to crawl on this web. So this one is extremely efficient. We never used it because you put it one female and then the whole population is like down. These ones are very generalistic predators that can feed even on pollen a little bit. So I don't think they compete a lot for the space, but mostly for the resource.
Okay, I've got there's really some quite questions about the immune responses of the plant and reading the thesis.
Yeah.
You mentioned that like JA, SA and ethylene are the ones that everyone knows about.
Yeah.
You know, these other ones which are still important but people know less about. Is this because they are less important, or have they just been less explored? And you mentioned that you've worked on it a bit, but you didn't elaborate that much.
Okay, that's a good question. I would say that salicylic acid and jasmonic acid, it's a kind of an historical thing, where they found these two different phytohormonal pathways that were cross-talking. There was a trade-off, and for many years, even if we know that there were auxins, and then we had also mutants, tomato mutant plants, where you can block these two responses, so it was practical. And I hope I'm getting it right, but I think that at the beginning there was all these secondary pathways that are also sometimes dependent on these two main ones. But again, it depends on the system. If you take tomato, there's these two different pathways, but in some other plant there's other pathways that play important roles. But for example, cytokinins, which are important phytohormones, I think they are by far less important in all this kind of induced response of plants, emission of volatiles, and etc. So, to answer your question, I think they are important. They are more important than the others, and of course they have been more explored. But it's not the end of the story.
Okay.
Yeah.
And for the thrips, you think that most of the symbionts are environmentally obtained?
Yeah.
This is kind of going on from what you were talking about with focus.
Yeah.
Will this just be? Is this? Yeah. Why do you think this?
Tough question. I think that, so we did the analysis so we did replicates for each PCR, so we know that someone who is there is there. We don't know the abundance. These are small animals, so the amount of bacteria that can fit on these of the animals is kind of limited. We had some preliminary studies doing this sequencing of specific thrips, and they found also quite a large diversity. And they did this previous study. They also did some kind of functional analysis of these different bacteria, and many of them, if I'm not wrong, were just passengers of these insects. I think that the work that we need to do there is to go into the detail of the different species, trying to blast them in databases and try to find information about this kind of bacteria. As I've said, if we have a Wolbachia, a Burkholderia or Hamiltonella, this is likely to be symbiotic. And then there's a lot of environmental contamination. Maybe sequencing some flowers would be a good way to get rid of some noise. That's something that definitely we need to do. Again, to answer your question, it is very difficult. I think that, okay, I would say that 20% are maybe environmental.
Okay.
That's my guess. We don't know nothing about thrips. It's so unknown, and it's, yeah. 25.
I was just wondering, how starting working on with Flo with the Wolbachia, so starting to think about it a bit more. And again, coming back to the thrips, so you're saying that maybe it's like a positive, like pollination, so it could be like a positive effect for the plant.
Yeah.
But also it's like it's a huge. They damage the leaves, don't they? Because the larvae are on
the plant. Yeah. That's a big. That's an important point. So what we sample is adults. When we sample these adults from flowers, we find little larvae. Most of the insects that we get, they are adults. Now that we are screening the tubes, there's few larvae. So I think that my guess, so there's some evidences of thrips which are specialist pollinators, that there's a very intimate interaction with some groups of plants where the thrips is the main pollinator of these groups. These are obligate pollinators, but from all of these species we know little. There's a big group of thrips, and the larvae feed on fungi in the soil. These larvae feed on the fungi, the adults get out and they go to flowers to mate and to get some pollen. So we don't really know. With the paper that we published with Niri on the elevational gradients, we saw already a very strong structure of the thrips community in relationship with the flowers. Meaning that this initial idea that thrips, they are dispersed by wind, they drop in a plant and they go to another plant, that's not true. These animals, they select their plant, they can disperse more than we think, they are kind of picky with the resource. From this to say that 50% of the species are pollinators, that's my guess is that they go there, they feed on some pollen, they mate, and they can take a bit of food from the flower, maybe picking up from the leaves.
One last question. Okay, I really like your the mentor first thing, and I sent it around our diversity group at ESM, and we're wondering about how to make training obligatory for mentors or people who what do you think about this, and how can we implement this when apparently this has to be imposed by our tutors, not by...
Yeah. Well, we discussed about that yesterday a little bit during dinner. I think that there's a kind of a movement where all our institutes are putting in place this kind of system of gender balance, and even for the juries you have to have a kind of a parity. I think that you have to put this kind of initiatives there, but you need to reinforce them. If there's cases of harassment, you have to do something about it. And we see too often cases of really ugly harassment that goes like this and there's no consequences. I think that there has to be consequences, and if you don't treat your students well, maybe you cannot supervise more. For me, it should be like that. But of course, like all changes, we should take less the plane, and we do it. But I think that if we think that our institutes, we should change from talking about it to acting about it, even if you have a researcher with a lot of millions, it's not enough to justify some behaviors.
Thank you. So I'm the last one, and I think we have time at the end if you want to think about one last question that each one wants to ask. I'm gonna give you the opportunity. So thank you for the presentation, for the rapport, and for inviting me here. I appreciate it, as everyone else said, the way you presented things, and I also appreciate the conversations we have regularly in the lab, because I think you put out what you're writing. You're putting out a good work environment and a scientific environment, and that's highly appreciated. And I wanted to start with a challenge. This is going to resonate with you, Elodie. So I was reading the report, and I was finding words that ennemis naturels, lutte biologique, défense, antagonistic interactions, enemy cocktail. So if you're talking to a journalist, one would think we're in a war against a portion of biodiversity.
Yeah.
So my challenge is, can you define biocontrol without using a vocabulary of war?
I would define biocontrol as a mutual agreement into two different trophic levels, where one decides not to consume a lot on the other trophic level for the good of the plant. I don't know.
So it is a difficult, difficult challenge, but the way we talk about our science actually has an influence on the way the general public thinks about our work and about biodiversity in general. Having started with that.
No, that's a good. Maybe the next one could be like the agreement project or something.
So I have just a couple of scientific questions, and then I think I'm gonna try to go towards a different topic. But you mentioned the counting is difficult and takes a lot of time. Do you know of any efforts, or is there a possibility to somehow automate counting using photos and artificial intelligence? Do you know of any?
That's my dream, to have the cages running automatically with one little camera. So that I thought about it, and I discussed about it with some people in the lab, to just, for example, take pictures, and then even with the pictures you can even catch interactions. You see this ladybird feeding on a parasitoid, for example. That would be great, because of course, if now we had 400,000 counts, we could have like even more. The problem I see from what I discussed with people is that our cages are too complex at the structural level to put a camera that can count for this. It has to be a flat surface somehow. So.
But when you're counting, what are you counting? Are you counting? Are you taking a leaf out of the plant?
with the needy, we were taking a leaf out of the cage, even if I told him not to. So this way I let him be free to choose. I told him, like, it's going to be insane. He spent 6 months counting and measuring square centimeters. He wanted to do that. It was good with me. The data is very precise, but I think that this precision was not justified for the amount of effort. So in that case, that could be possible. And maybe for the experiments that we are starting to launch with the spider mites, that could be an option, because the only choice we have with spider mites is to take the leaf, put it flat and count in the microscope. So for that would be potentially doable. I contacted a company that does that. The price was quite insane to put in place to do this training of the model. And the other problem we have is that it's not only counting spider mites. We are counting different species of predatory mites. Some of these predatory mites distinguish. There's one of the 16 combinations of enemies where we have 2 predatory mites which are very similar. You have to take them and look at them in the microscope to count 2 little hairs in the back of the insect, of the arthropod. So I think that we are not at that point yet. But that would be extremely nice, because then the students could dedicate their time at doing real science and not only counting. But so far we are not there.
Okay, switching gears a little bit. So you talked about the biodiversity biocontrol link, and that reminds me a lot of the biodiversity ecosystem function relationships.
Yes.
And of course that brings to Tillman grassland experiments where people have been manipulating the number of plants that you put on a small square of land and changing the identity and number of plants you are measuring at the same time, some sort of function.
Yeah.
And in those grassland experiments, there is not even trophic interactions. It's plant diversity versus ecosystem function.
Yeah.
And when you go to 6 to 8 and maybe 10 species, the relationship is usually increasing, although it can be idiosyncratic because it depends on the identity of the species that you add. But then things can get more interesting as you get more than 10 species. And in some of these experiments that you are doing, in most of them, you have in general less than 10 species, I believe.
Yeah.
Do you think that you're missing an important part of the dynamics there? And in particular, because when you put this in a real system, there are going to be species that come from outside. So what could you be missing? And is there anything that you can do to deal with that?
Yeah, I think that we are missing a lot. Mm-hmm. I mean, the way we plan the experiments with combinations of 2 enemies. So I talk about cocktail, but it's only 2 enemies, what is written in the project. So it's very limited, and it's far from what is the real diversity. Ideally, if for example, if the PhD goes well and the initial experiments are quite encouraging, we could add more species, especially more species at the plant and the insect level. So I think that we discussed that yesterday having dinner. I think that one of the reasons why these dynamics are not stable is because there's only one resource. There's one sweet pepper, one aphid, and it's like I put always the example of a grass, and you pass with your with your machine. It's very easy, like you just clean everything. But if in this, it's not a golf can, but some plants, then you need to change the knives below, and then it can be more challenging to kill everyone. So I think that we need to add more plants in the system, maybe, and more aphid species, maybe more genotypes.
And so the other sort of theoretical aspect that is very often mentioned is that there is a trade-off. That is, it starts from a theoretical prediction, but it has been shown in real life. There is a trade-off between being a really good competitor versus a very good disperser.
Yes.
And so a poor competitor can escape.
Yeah.
from competition by dispersing.
Yeah.
In your project, do you have any sort of setting, experimental setting that would allow you to look at these spatial dynamics a little bit?
No, that's another important limitation of the approach that I presented today. It's only based on communities, not meta-communities. And that's a big chunk of research that is missing. We could do that a little bit by connecting cages with tubes. That's something that I've thought about many times. But we have a full rearing room with 40 cages. If you connect them, you end up with 20 replicates only. So I think we are quite at the limit of our technical possibilities. One other thing that when I try to think about the next level is that we have some field sites over there which are kind of empty. I would like to put some mesocosms, like these 2x2 cages that Dirk Sanders works to just put the communities there with a lot of spatial structure, many different plants, and a lot of hyperparasitoids that will contaminate, because there's always this problem.
Don't you have a component where you're going to be releasing these sets of communities in a larger space, in greenhouses?
Yeah, of course.
And are you going to follow the dispersal? Maybe? Do you have planned any settings?
It is not planned, because the way we have thought about these experiments is to have our greenhouse compartments. We will sample like 10 plants, as we did in Reunion, for all the natural enemies and pests in these 10 plants, and mass release and inundate these greenhouses with enemies. How could we measure dispersal? Well, one way to do it, and that could be quite exciting, would be to put, for example, outside, I'm just speaking up my mind, and putting it outside the greenhouses, putting some sticky traps to see how the enemies escape in some situations where there's more predation risk or intra-predation, for example.
I would imagine a scheme where you don't introduce the predators in randomly in space, but you put them in some place and follow how they spread, for example.
It is doable, but if we manage to do the first part, we are already superheroes. We have to manage to get the company to produce them, to find the replicate greenhouses with aphids.
Yeah.
But I'll let the later.
In terms of how to bring this in the field, so I don't know if you've seen the new draft of the Ecofito 2030, or even the old ones. Biocontrol is really at the center of how the government plans to replace pesticides, although the definition of biocontrol is a lot larger.
Okay.
Yet, regulation in the EU to implement a lot of these new strategies is very heavy compared to other countries, other places in the world. So
heavy in which sense?
Heavy,
that is difficult to implement, you mean?
There is a lot of regulation
okay.
In terms of how do you go from designing any kind of non-pesticide or even pesticide, but non-pesticide solution to put it in place in the field.
Okay.
So I was wondering if you've thought about or encounter regulatory barriers to developing this sort of or implementing this sort of biocontrol options.
Not so far, but I have to admit that I've been quite behind the application. So when I showed this data, that was the closest I did in terms of transferring to the applied, just providing some evidence, doing some PhD projects, some master projects, and saying, hey, this is the evidence, see what you want. For the project that we are now developing, as far as I know, there's not much problem, because the way we think we are going to do that is to talk to the farmers, telling them, let us release some enemies in your greenhouse. If you need to put insecticides, let us know. please treat them all equally. And but there's no regulation problem because these are all indigenous species. So technically, these are species that are on the market. They are all indigenous. There was Harmonia, for example. I wanted to put Harmonia in the project, and the guys in BioVest told me, like, you cannot put on a species that we don't use anymore, and it looks really bad to put this in our project. So I removed it. That's some of the conflicts when you work with a private partner.
If I may say, that's one of the advantages of this approach. You're working with the menu of already approved.
Exactly.
Because I think that will require very little extra regulation. But just to put a single other beneficial onto their menu would be a huge cost and effort in terms of regulation.
Yeah.
And so my last question is, can you go back to the slide with the tweets about mentoring, putting the priorities first, the student priorities?
Yeah. That one or that one?
That one.
Yeah.
Okay, so you have four suggestions.
Yeah.
But in concrete terms, how do you implement them? What exactly did you do, for example, to recruit your PhD students, or in your ANR meetings, or you know?
Well, I think that well-being, that's easy. Well, okay, let's go about the, for example, the parity in terms of how I do the recruitment. Huh?
Mm-hmm.
So,
or put the question in another way, it's like, what would you recommend people to implement systematically in their own working groups?
Yeah, well, there's always the question of diversity in your groups. So with the PhD with Francisco, we got 120 CVs. That was tough. I wanted to do this blind analysis where you cover the origin, the sex, and all this from candidates. That would be the ideal way to do. With 120 CVs, I would still be doing that. Mm-hmm. So what we did, I put a panel of 3 people. Already with some parity, there were some women, some men. I did a pre-selection based on the cover letter, trying to not have any bias, and then we analyzed it with the whole team, trying to avoid these biases. One thing that I did quite actively when I was in reunion, that it was a team with 2 PhD students, 1 technician, and me, which were quite men. We asked we wanted to recruit a master student, and we eliminated almost all the men, and we just focused on CVs by women. And when we found that there was someone that could do the job, then we were happy with that. Of course, if out of all these women there was no good candidate, then we would have moved to the men. But this was one action that we did. We want to recruit to put some more parity in our team, and we did that, and we hired a lot. So I think that these kind of initiatives are useful. In terms of co-authors, as I showed, it's not always easy to choose your collaborators based on sex, of course, but it's good to. I think that when I was a student, I had this state of mind that good science came from men, right? I mean, it's a bias that we all have there, and I think it was influenced my way of my career until I realized that, and then I tried to get rid of this kind of bias. It's not always easy, and we do what we can.
And you said promote self-care and well-being. That's easy. I don't think it's easy, so I would like to know what you do to do that.
I don't know. What do I mean by well-being and self-care? I just think that you have to think about try to see how the people you are working on, how they feel. Ask about their life. Are you okay? Do you like this experiment? How do you feel? Do you want to talk? Do you need a break? Take your vacation? You work too much? These kind of things. I mean, I think that this is something that you have to do, even if you are extremely busy sometimes and you don't want to have this kind of talk because you have to do your analysis, but it's a matter of putting effort on it.
Good. Okay, so do people have their additional questions?
Okay, I have a very selfishly interested question.
Okay.
Because you've been doing like experiments very heavily with a lot of work into it, but you also like publish so many reviews and meta-analyses.
Yes.
So it's like incredible. And I mean, the colleagues I've been talking about with lately, everyone tells me that they can't keep up with the literature anymore. There's too many of it, and we have so many tasks to do, and no one manages to keep on track with it.
Yeah.
But you seem to do it, and I want to know how you do it.
I think that about the reviews, I learned a lot when I was postdoc, working with people that were quite good at writing. I learned a lot from them. And about the reviews, I think that this is something that I enjoy a lot. So we are working on a review with Dirk on ecosystem engineers and trophic cascades. I knew nothing about it, but I got into Web of Science, started downloading papers, and I enjoyed it a lot, reading, even if you just read the abstract. And of course, then you go to the details if the paper is relevant. But just reading abstracts and seeing the idea of a field, I think that it's something that is doable, or at least I can do. And then some ideas pop up, and I like it. I like it writing reviews. And
are you a very fast reader, or do you have a method how to screen a paper super efficiently? I don't know. I want to know. Give me your secret.
I'm not a fast reader or fast. Twitter helps, I have to admit. And I don't write very fast, but I enjoyed it. And I saw it when I was writing the HDR. I was quite like, I'm going to be able to write all this. And then I started, and it went pretty well. But there's some initiatives. For example, I have my friend Ana Pineda, who has some courses about it, and she talks about how to deblock writer's block, how to plan your, and I have some, for example, for the HDR, I was using a lot Pomodoro, which is a technique where you block like 30 minutes of writing, and then you disconnect your phone, you disconnect everything, and you have half an hour. And then what I was doing is like, from 9 to 12, I need to at least do 2 Pomodoros. It means it's 3 hours, and out of the 3 hours, you write concentrated 1 hour. And you say, that's very easy. It's not easy, it's very difficult. But if you have the timer there, you do it. And it helped me, but I kind of, yeah. It's a very nice comment anyway.
Elisa?
In fact, I have too many questions and nothing very general. I guess one question I wanted to ask, but it's more a thought, like when you analyze elevational gradients, you said that it was interesting in a climate change context. And I was wondering if increasing temperature, like your hypotheses are between harsh and not harsh environment.
Yeah.
But here the harsh is more the cold side. And I was wondering if it was a good,
Yeah.
You see what I mean?
I see what you mean, and I accept the comment. I want to add something to your comment before. I haven't done it on my own. I work with a lot of people, so that the credit goes to the others too. Okay, that's important. That's part of the well-being. I think that word harsh is not very correct, because especially thrips that loves being at living at 1,500 meters, that's their environment. What is harsh for this individual is to go to the tropics. I think I would limit it more to the amount of resources available. So I think that at high elevation, low temperature, less productivity, this is clear. Now that I see that, I think I should have replaced the harsh condition by less a lower number of niches available.
Yeah.
And I think that this is correct. There's less plants, so yeah. So that's a good point. Yeah.
Right.
Maybe a question on risk taking and failure.
Okay. Yeah, that's a good question.
Because because actually I'm really fascinated by the risk you take when you do these kind of experiments.
Yeah.
And so I wanted to know what you think about that, how we can handle that, and how we take it into account, especially in training.
Yeah. Well, the first thing I always tell to the people I work is that failure is the rule. So most experiments will not work. I think it's good to have in your menu some risk experiments. Like when I was Marie Curie fellow with this volatile thing, it was an extremely risky experiment that it went well, and it gave me probably the job that I have now. But in parallel, I did many other experiments that some of them worked. They were less exciting. I never managed to publish them because I didn't have time. So I think that this is a good balance. With the PhD that we are launching now, for example, I think that there's some basic, to get a couple of papers on biocontrol journals, I think it's going to be relatively easy, just by doing experiments and saying they're very effective, they suppress the symptom. And then I always tell the students, this is a very risky experiment, there's a lot of effort. If it works, you can get a very good paper, measuring it as an impact factor. That's a bit a pity, but that's how it works. And with this good paper, you can have the chances of getting into a postdoc. So try to balance these different things, and then, and of course, launching several things at the same time is a good way to balance the risks. And I don't think the project is very risky. No, yeah, it's a bit risky. But yeah, and of course, being ready for frustration. I look like I'm super easy, but when I get a paper rejected on an experiment that doesn't work, I'm not happy. I'm just feel miserable.
We are discussing with Denis about registered reports.
Registered report?
Yeah. So the thing that you think about a project,
yeah.
You submit the protocol of the project, yeah. The idea, the protocol, and so whatever the results you get.
Yeah,
you can be able to value the experiment and the trajectory, and maybe that's something we should think of, because then it means that okay, you can take the risk, because what we are looking at is whether you do good science and not whether you have good results.
Yeah, I totally agree, but we are so trapped in this system that you don't get an INR if you don't have good papers. I mean, why not publishing the non-significant results? There was a journal, the Journal of Non-Significant Results. It never launched because no one wants to publish these results because no one validates them. And it's a pity. For meta-analysis, it's a big pity because we do meta-analysis, we only get the significant results. Whoa, super significant, but...
and we invent methods to correct biases in reporting results. So,
but then in systematic reviews to now often do that. They write a first article, how the systematic review will be done, how the papers will be selected, and that's one publication. Only then they actually start doing it.
Ah, yeah.
That's an interesting
maybe
approach.
Could be an interesting approach.
But who wants to write 2 papers?
The registered report might be a very good idea, because you publish the idea, the protocol, and then if you are reviewed correctly on that basis, it means that the results will be published.
Exactly.
Christophe, you want to follow up with a question?
I guess it's more a comment, but I very much agree. Your general conclusions are right. If we want to move towards a more sustainable agriculture, we need better uptake of organically produced produce, which requires a drop of prices in these products as well. Mm-hmm. But even releasing a single natural enemy is much more expensive than chemical control. So, in a way, what you're advocating to use these cocktails, I can see how it makes biocontrol better, but I'm not entirely sure it makes it cheaper. So, how can we make it better and cheaper?
Well, I mean, the easy answer is that with our great knowledge, we will make it more efficient. That's the way that I could sell it. The difficult answer is that it's not only about prices. I mean, it's also about how we consume things, where we put the money in our life. So, yeah, I would stay with a simple question, which is, I think that with more knowledge about these enemies, then we can make it more efficient. I think that what we get from these biocontrol companies is still the tip of the iceberg of what we can use. And this comes to the comment by Alison. These companies, they sell you this same genotype for all the problems. This generalist predatory bug, Nesidocoris, is used to eat tuta absoluta, which is a moth, aphids, spider mites, everything. We could do it a bit more adapted to, like, you have this natural enemy specific for tuta. But then, of course, then it goes to the benefit of the company to produce it. Call me communist, but if this was managed by the state, maybe that would be more easy to manage all these collections of different enemies that then we could deliver more easily. Of course, the economic situation is not going in that direction, in France at least, but that could be a solution. But, yeah, a very romantic answer.
You can start your company, BioBetter.
Yeah, BioBetter. Yeah.
I haven't really got a question, but sort of something, I'm going to throw something back to you that we've kind of discussed and you've asked before, is like, you refer to these as herbivores, and I refer to the spider mites as parasites. And like, herbivore to me is more like a deer or a cow.
Cow, yeah.
Yeah, so...
Yeah. Yeah, we had this discussion, especially in French, where with my abstract in French, they were like, herbivore, that's a cow. I always thought about insects are herbivores. I mean, if you think about our planet, the ones that control primary production are mostly arthropods. So, well, maybe it's not true. In the Great Prairies, the cows and the mammoths and all this, they did a lot of work too. I don't know, I think that this is something that, for example, in Wageningen, which is a big lab, a huge lab working with insects that feed on plants, it was herbivores all the time. No one had a problem, and I adopted it. So this is maybe my past. I think that you are in the field of more evolutionary ecology, and then it's like the biocontrol way to talk about biocontrol. There's many different definitions, but in an entomology conference, you talk about herbivores, everybody thinks about the cricket, so that's fine.
In the food web, it's the same. We call them herbivore.
Yeah.
Yeah.
And so my last question is, do you think, or are you encouraging your students to publish in PCI?
Wow. I think I will let the decision to them, but I will tell them to be careful about which type of career they want to do. If they want to stay in France, PCI is totally fine. If they want to go to the States, PCI is likely not to be enough value for the Americans. I hope and I wish PCI will become a reference. That would be the perfect scenario. In the short term, for a PhD, for a master, for a PhD student, is a bit risky depending on which career path this person wants to. And even myself, I still feel like I will still have some problems in getting all my papers in PCI, because we look for international recognition at some point to get projects, whatever, and
Even to get you.
even people that tell you good paper Frago in the conferences, and this is achieved in other journals. I think I will discuss about it, and I propose this menu of options, and they can choose, and I'll be happy to help them with that.
Well, thank you. I think we've done the rounds And so it's the moment where the jury will convene and discuss between ourselves.
Okay.
Do you happen to have
I'll close the...
Yes, we need to go to the restroom too.
And thank you very much to everyone, especially the ones staying until the bitter end.
Bitter end.
Merci à tout le monde en ligne. Je vais couper le streaming maintenant. Adeu pares, si encara esteu ahí i heu aguantat fins al final.